Welcome to SRB026. What are you, Dave?
That was easy. Okay, well, if we could just have the agenda brought up, please. Transitioning back. Could we just have the agenda up, please? So for today's agenda, so we have essentially 2.5 days, 9 to 5-ish today, the usual drafting session in the afternoon if you want or need it. And then just a half day in 12, where we'll go ahead and go to the report. In terms of the agenda itself, it's just the standard SRB agenda for the June meeting. We have the IPHC process components, and then we'll run through the usual updates. And I think we have some pretty interesting updates from Giuseppe's team in particular this meeting. So I'll pause there and just see if there's any other requests for additions or modifications to the agenda before you want to adopt the chair.
Adopt the agenda.
Okay, we'll consider that adopted and we'll jump straight in. Just a quick apology from Anne-Marie, who isn't able to make it in person or online for this meeting. And so during her observer updates, I'd asked if she had any particular updates. So nothing new from last meeting. So in that sense. Okay, if we could have first paper associated with agenda item 3.2, which is the updates on actions arising from the 25th session of the SRB. And while that's being brought up, just a reminder that in terms of the annual SRB workflow, this meeting is designed for you to review the materials that are put forward, provide requests and redirections to the Secretariat to undertake an additional or modified work activities, and then to formulate your specific recommendations to the Commission at the September SRB meeting. The only caveat to that is, and you'll hear when Dr. Hicks presents the harvest strategy policy, there may be a good point in time to provide recommendations on the harvest strategy policy elements to the Commission earlier than usual, because they're looking to potentially have it adopted by the interim meeting in December this year. Okay. So in terms of paper 03, this is the standard update on actions arising from the previous SRB meeting. I won't go through the cover details, they're all the same, but in Appendix A, you will find all of the recommendations and requests that arose from the SRB 25th session. And throughout the course of this meeting, you're going to receive specific, more detailed updates. And so this is really just a reference document at this point in time. One thing I will notice, again, just reminding myself and reviewing this paper this morning, there's still a lot of work that's in progress that arose from that last meeting. The SRB was very active in terms of its requests and recommendations, additional work to be undertaken, and a lot of that is still underway. And so as I said, you'll hear about those specific updates where it's been completed, but then also if there's work that's being undertaken. So just unless there's any specific questions, we can just hold tight on those presentations when they come up. And if that work is still underway, it's just a reminder that we'll need to move those incomplete pending recommendations and requests to this report so that they don't get lost. And you can modify the report. So I'll stop there. Any questions on this paper? Too easy. All right. Well, thank you very much. Let's bring up paper 4, please. Lord, can I get you just to get escape on the keyboard in front of you there? So paper 4 is just the standard again, outcomes of the most recent commission meeting. So the 103rd session of the IPHC annual meeting, which was held in January of this year, and provided at Appendix A are the specific requests or other paragraphs or comments of note from the commission for the SRB's information. The primary one was that the commission requested the Secretariat facilitate informal intersessional workshops regarding the draft harbour strategy policy, and Dr. Hicks is going to walk through that progress a little bit later in the meeting. So we've held that first of probably 2 intersessional workshops, and they've received the draft, and we're in that planning stage of holding a second to take into consideration any recommendations or requests from this meeting into the finalization of that draft harbour strategy policy. There were 2 other, I guess, notings of the commission. The first was regarding the updated 5-year program of integrated research and monitoring. And the commission is asking that that be provided draft format to the commission by IM 101. Previously in November, but they've just moved the dates, and so it's now a 1-day meeting on the 2nd of December. And then the final, I guess, noting is the information paper, and Bush is going to present to you as part of paper 10 for this meeting. Again, a little bit more of an update. The Commission's very interested in how that work's been progressing. I'm sure you are as well. So that's it from updates on actions rising from the Commission meeting, unless anybody can remember something that we've missed in the paper. Word by word.
Yeah, can you talk more about that first bullet there, the MSC? I think we had talked about having Sean or someone else, the secretary of the MSC. Is that what happened?
No, Sean was unavailable for that. I'm trying to remember if you had a CCSPT or something, but I suspect they're going to, and then we'll need to try and make that happen for the work meeting potentially, if you're available. I know you weren't available for the work meeting last year.
That was in September.
Yeah, 4th, 5th of September.
4th and 5th.
I remember there's another meeting there for you. But when we have the second intersessional workshop, I think that's a good reminder actually to Sean join.
I'm not trying to make more work. We had discussed that we seem to be like having telephone conversations with the commission. Or this one, that's everything.
And I think with the discussions that Dr. Hicks is going to present on overfishing and overfish status, that might be a perfect definition rather. That might be the perfect time to bring Sean in as well as a chair. Alan, do you think anything else worth adding?
No, I do remember last year, it just didn't work out that we couldn't get the commissioners and any of the SRB members together. But yeah, the work meeting would be an obvious good place to do that, I think.
Yeah.
And that's in Bellingham.
Impacts that week. So 3rd and 4th. If I left on the 4th, I could be back here for the 5th. I don't know where the meeting is.
In Bellingham. Is it the work meeting typically?
Thursday, Friday. It's usually just a 1-day, Thursday, a full day. Similar here.
We haven't scheduled a workshop with the commissioners yet though, either. I don't know if that's gonna happen. But we'll keep it in mind and we'll bring you in.
Might be worth doing it earlier. Okay.
It's nitty gritty.
Okay.
Yeah.
That closes that agenda item. The next is just the update from observers. So he hit escape again for me, sorry. Paul. And so as I said, Anne Marie is unavailable for the meeting, and I asked her if she had any additional updates or questions for the SRB or comments from Canadian commissioners. She indicated nothing new at this point in time. And so with that, Pete, are you online and have anything for us?
Just got an email from about sound or something. For some reason, the sound just dropped from the SRB meeting. And there's no chat available, Pete says. I don't,
You might have to try and so he can't hear us at all.
he just sent an email. Yeah, it says the sound just dropped and he wasn't able to chat.
Microphone looks okay.
Yeah.
Try and make a represent and see if that works in.
Just in 9:12. That was, yeah, it was a minute ago. It might have just dropped for some reason.
I don't know if you can hear us, Pete, but I've just made you a presenter to see if that helps, but your microphone is not showing anymore.
I'm just texting.
Where's Pete based? He's here.
You know, he's in Juneau.
He's gotten the chat to work now.
Oh. Yeah, he says he can't hear us.
All right, Pete, I know you can't hear us, but just for those others listening in, I guess, we're going to try and work with you to try and get you connected. And then you can chime in with observer updates as soon as that happens. Right, I see you're going to try and reload the meeting, so that'll be great. Okay, so we can move on and then come back. That's okay?
Sure.
Okay. So that will take us to agenda item 4, the IPHC 5-year program of integrated research and monitoring. We don't need to bring this paper up, as it's just the same as last year. And so if you recall, the SRB had requested that we front load the revised or updated 5-year program of integrated research with the key outcomes from the previous 5 years. So that's just a work in progress at the moment. And we're going to need a bit more time to really develop that into a new 5-year program, integrated research delivery from 26 or 27 for the next 5 years. As I was mentioning in the outcomes of the commission, they would like to see that draft by the IM, so 30 days prior to the interim meeting. It's about the 2nd of November will be a 30-day deadline. That's our core target. So at this point, we're just planning on sending that to you probably earlier than the 30-day deadline for the next SRB meeting, so that you can try and review that and provide some additional input that we can agree to for the SRB meeting. So a bit behind in that sense, but that's the plan for the update. Any questions? Good. Okay. If not, then we can jump into 4.11, which is under research, biology and ecology. So Giuseppe, over to you. Which presentation number is that, Giuseppe?
That's presentation 06.
Which is presentation 06, please? Can you operate from there or not?
No, can I be made presenter, please?
Go
great, thank you.
Pete did say he has sound back.
You want to take Pete very quickly? Before we jump in,
Yeah.
Pete, can you hear us?
Yes, can you hear me?
Yes, we can. Welcome. Over to you.
Great. I always gotta have some sort of technological glitch at the beginning of a meeting. Well, thank you so much for the opportunity to ask questions on behalf of the U.S. commissioners. The questions that I got from them generally dealt with two categories pertaining to this year's assessment. The first one is with the updating of maturity. And it's kind of a multi-part question, but it's essentially is around whether or not the Secretariat has investigated relationships between changing maturity and size and age over time. And if so, or if that's in the plans, perhaps how that might affect the assessment outcomes going forward.
Second one?
All right, I guess that's a stumper.
Let's get them both and then decide who's going to answer what.
Okay, sure. So the second one has to do with the FIS and essentially dealing with all of the missing areas in last year's and this year's FIS survey design. And the question being whether or not it's being considered to use some of the fishery dependent data to inform abundance trends within these areas that are missing. And if so, and whether really if that is going to be considered for use in the 2025 assessment. And those are the two category general questions.
Good. So I think probably, Joseph, can you address the first one in your presentation? Sounds like you could do that.
Yeah, I'll probably need some assistance from Ian as well in that regard, but as you'll hear just in a minute, we're going to be providing some revised maturity estimates based on 3 consecutive years of data from 2022 to 2024. And it looks like from what Colin will just indicate in a minute that the maturity schedules may have changed with regards to the ones that we're currently using for stock set. How that relates to changes in size and age, that's an important question that we are not ready to answer right now with the data that we have. We suspect is that there may not have been so many changes relating maturity schedules in relation to length as opposed to age. So that's a relationship that I think it's worth investigating. So far, we really don't have a good grasp as to how those changes in maturity may have been taking place in the context of changes. Again, do you want to add anything else?
Just that the big changes in size and age occurred over a long time period that we don't have maturity estimates. So the real action, we don't have data right now. I think looking forward, that's going to be one of the main, that's probably the main focus of continuing to monitor maturity would be to be able to do size and age. But obviously it's going to take years to do that looking forward. I can segue from that into the second one with regard to using fishery dependent data.
So
Sure, but
first one, I have somewhat possibly a naive question. Does anybody know if there's any kind of signal in the otolith of maturity? Is there a change in growth, a concentration of growth when they mature?
Not that I know of. Yeah, it's plausible, but I don't think I've ever seen that documented. Most of the change when it comes to like otolith rings is based on pelagic to the benthic environment settling out. That's a big change, especially in early life stages. And then obviously as you get to annual rings, they can change through time based on environmental factors and those things. But I don't know anything as far as in the literature regarding that type of analysis in the otolith.
So on the second point, the question about using fishery dependent data as we have greater uncertainty and or missing areas in the FIS, that's essentially exactly what we've been doing in the last several years. So unlike some assessments where you might have only a survey index in the stock assessment, we have both the survey and the fishery CPUE index in the stock. And so as the variance of the survey goes up, naturally we are weighting the fisheries CPUE more heavily, because in the extreme, if we had an infinite variance on the survey index, we would have, the only index we'd have would be the fishery index. So the model would inherently fit that better. I think that's a cleaner way to approach it than actually trying to blend the two outside the model into a single index and then not really knowing which contributions were which and how to interpret that information. So we are definitely doing that. And as you'll see when we get to my assessment presentation, we're seeing some slightly different signals between the two data sets. And, but we are in the last couple of years increasingly fitting the commercial fishery CPUE index more so than the survey. So I think we are accommodating this effect of missing survey areas, and we're definitely seeing the effect of the assessment. The uncertainty there is whether or not we are now, out of necessity, fitting to a CPUE series that may be reflecting other information beyond just the trend in the population. Because we do know there are other factors affecting the fisheries.
Or is that an indication of emerging bias? Something we're concerned about as well.
And those are the two potential hypotheses, which unfortunately, without better survey data, it's difficult to delineate. I will say that the survey that's already underway for this year, while not as comprehensive as you'll hear more about it, while not as comprehensive as the base block design that we would like to see on an annual basis, is quite a bit larger than the survey we had in 2020. So I expect better information this year than we've had in a couple of years. We can loop back to this when we get to some of the specifics of the stock assessment as well.
Okay, Pete, how do you like those answers? Is that enough?
Yes, that's great. Thank you so much.
Bill, sounds like you'll get more details on those in the individual presentations as well.
Absolutely.
Okay, back to Joseph. You can continue.
Yeah, thank you. Good morning, everybody. It's a pleasure to be able to provide an update on system science research activities. The presentation today will focus mostly on updates on two areas. One is population genomics, indeed, just a minute, and then following the presentation by Colin regarding the update of maturity schedules, which just discussed. We'll be providing also very brief updates on some of the other areas that we're going to do. So the first area that we're going to be providing some updates on is the research area that we titled Migration and Population Dynamics, and this is the work that we've heard in the past from Andy related to the establishment of a baseline of genetic diversity and the delineation of fine scale stock structure. What Andy will be presenting just now is really an update of those analysis, incorporating additional samples. And hopefully this will lead towards the completion of this project as it was presented, and we'll be working on final review and hopefully an important peer review publication. So I'm going to pass the word to Andy, and you can change the slides directly.
All right. Thanks, Josette. So yeah, as Josette mentioned, this is just an update. So since the last SRB meeting, we conducted some additional sequencing. And so this next few slides are going to be an update on how the additional samples that we've added to the data set affect the results of the core analyses that we're basing our main conclusions on. But before we get into that, I'd just like to start with a little bit of a reintroduction to reorient everybody to the study design. So the main objective for this project is really to resolve the genetic structure of civet halibut in IPHC convention waters. And the method that we're utilizing to generate the raw data for this project is called low coverage whole genome resequencing. And this method allows us to screen genomic variation at very high resolution. Essentially, we can sample every single base pair of an individual's DNA using this technique. So the additional sequencing run that we carried out was done on Illumina's NovaSeq X Plus platform. And with this run, we submitted an additional 384 samples for sequencing. And the primary purpose for this sequencing run was to increase and balance the sample sizes for our baseline set of sample collections. If you recall, the very first sequencing run we had some issues with, and we chose to just remove those samples from the analysis. So this is really an attempt to recover those and bring them back into the data set that we initially intended on setting out to create. Also, this gave us an opportunity to sequence some additional summer collected samples with an emphasis on samples collected near the latitudinal extremes of the range of the species. So I've revised the map here to reflect these additional samples. And so the points in red are samples, the locations of samples that are associated with the baseline set of samples. These are samples collected during the wintertime when fish are assumed to be on the spawning grounds. And so we have samples from British Columbia, Gulf of Alaska, Bering Sea, and then the central and western Aleutian Islands. And then the points in yellow are the locations of samples collected during the summertime. So we have samples as far south as Eureka, California, and southern Oregon. We have some samples collected in the Gulf of Alaska near Kodiak Island, and then into the Bering Sea, extending into the northern Bering Sea. And then in the inset here, we also have Olaf was kind enough to collect some samples from Japan during his trip there last summer that we've also sequenced on this run as well. So as far as the bioinformatic processing methods are concerned, we utilize the same procedure that we have for previous sequencing runs with some minor modifications. So first, we removed individuals if their average sequencing depth was less than 1x. Previously, this was set at 1.5x. So we're relaxing the stringency on what we're requiring to include an individual in the data set here by a little bit. And then we also used a piece of software that's designed to identify genomic regions that are problematic for sequence read alignment. So you can think, sometimes you get duplicated regions of the genome or highly repetitive regions. And so these are great problems for read alignment. And so basically what we're doing is identifying those regions, and then any mutations in those regions are excluded from the data set. So we're being more stringent with we're increasing the stringency of what we're requiring to include a SNP in the data set or not. Okay, so as far as the results of this sequencing run are concerned, we observed an average coverage of 2.4x for the sequencing run. So that means we're sequencing an individual's genome on average 2.4 times over. And this run enabled us to include an additional 161 samples to our baseline data set. So now we have a total of 731 individuals in that set. And the kind of breakdown of the sample sizes associated with each collection are shown in the top table. So if you recall, previously we intended on sequencing about 50 per sample collection, and now those numbers are up near 60 per sample collection. And then also we now have genetic data for 327 samples that were collected during the summertime, and those numbers are shown in the bottom table here. So following, the initial analysis and the SNP calling procedure, we're working with 3.7 million SNPs for the downstream analyses that we're talking about today. So the first thing that we did with this data set after we added those additional samples was to conduct principal components analysis, because it's a nice tool to visualize any major patterns of structure that might be present in the data. And so the procedure here is to estimate a covariance matrix from the genotype likelihoods using a piece of software called PCANGS. Then we take that matrix and do the eigen decomposition in R. And when we initially did this, it was apparent that there were a few samples that appeared to be outliers that appeared very different than like the main cluster of points, and that made it difficult to visualize any patterns that might be present in the main group of points. So we used an iterative outlier removal procedure to deal with these problematic individuals. And what we did was we removed an individual if they were greater than 6 standard deviations away from the mean along one of the top 3 principal components. And we did this, and then we reprojected the data. And then we did this for up to 10 iterations or until all the individuals were removed from the data set. And this flagged 22 individuals as outliers. So this figure that you see here is generated from 709 individual Pacific halibut. So, you know, each point represents an individual fish, and those points are colored by the geographic area in which they were collected from. And then the circles represent 95% confidence ellipses. And much like before, you'll notice that we're really dealing with a single genetic group of Pacific halibut here, and there's a lot of overlap among the different areas that were sampled for this project. So to kind of validate what we're seeing with our eyes with more quantitative methods, we performed k-means clustering using the top 3 principal components. And then inspecting 2 measures of 2 model sport metrics, total within cluster sum of squares and BIC. The continual decay of these values as you increasingly partition the data set into larger number of clusters makes it difficult to confidently select a number of clusters that best supports the data. And this is often indicative that you're dealing with a single cluster that, you know, best describes the data. Okay, so following the PCA, we then conducted assignment testing. And essentially with assignment testing, you know, we're asking the question of how accurately can we assign individuals back to the geographic area in which they were sampled from. And so the procedure here is to define your reference populations. And so in our case, we define these by geographic area, since we're interested in spatial differences. And then we utilize a simple training holdout procedure to assess the accuracy of those assignments. And this is really kind of the most conservative and recommended approach by some prominent authors in the field. So we did this in 2 ways. So first we conducted a 50-50 training test split. So half of this individuals within each geographic area were randomly assigned to the training data set. And then the other half were retained for the testing portion of the analysis. The additional sample sizes afforded by this most recent sequencing run enabled us to test out another scenario where we used an equal number of individuals per geographic area to construct the training set, while having, you know, a large enough sample size with each geographic area to accurately estimate allele frequencies, and still retaining enough samples in the training set to estimate those assignment accuracies. So basically, the first thing you do with the training set is you want to select a set of SNPs that's informative, potentially informative of your, you know, populations that you're defining. So the procedure to do that was we first estimated pairwise FST among all combinations of sample collection areas, and then selected the top 1,000 SNPs per comparison. And we required these SNPs to be at least 10,000 base pairs apart to minimize the effects that you know, physically linked SNPs on a chromosome might have. After we did this for all of the pairwise comparisons, we then combined the lists and removed any duplicates. And this left us with a panel of 8,575 SNPs for the 50-50 training test split scenario, and 9,078 SNPs for the scenario in which we constructed the training set with 45 individuals per area. From there, you define your reference populations or reporting groups, which boils down to estimating allele frequencies for those geographic areas. And then we use the test set to essentially assign those individuals back to the reference populations. And since we know where those individuals came from, we can then evaluate assignment accuracy with a set of, new samples that the model hasn't seen yet. So moving on to the results here. So these figures are confusion matrices that show how individuals assigned to each population. So on the y-axis, we have the population of origin. And then on the x-axis, we have the population in which those individuals were assigned to. And then each cell has the number of individuals associated with those respective assignments. So the figures on the left are the numbers associated with the test set assignments. So these are the numbers that we're using to calculate those assignment accuracies. And then the figures on the right are the self-assignment rates of the training set that was established using leave one out cross validation. So as far as the 50-50 training test split scenario is concerned, these results are identical to what you've seen before and what we've produced with the reduced data set. We're observing about a 33% assignment accuracy, and all of these samples are assigning back to the Gulf of Alaska with very high probability. And what I think is going on here is that since the training set that we're using here has the largest number of samples from the Gulf of Alaska. So the geographic area with the largest number of samples is the Gulf of Alaska. If you assume that there's a single genetic group of Pacific halibut coast-wide, we're able to obtain the most accurate allele frequency estimates for the Gulf of Alaska. And those are probably most representative of the coast-wide group. So it's capturing all those individuals in the training set. And that's, I guess, yeah, that's what I think is going on here. Now, when we look at the scenario where we used 45 individuals per area to construct our testing set, this figure looks a little bit more interesting. However, the assignment accuracy actually decreases to about 27%. And in this case, we also observing about 8% of the samples are being categorized as unassigned. And so I think, taking the low overall assignment accuracies combined with the perfect self-assignment rates of the training set really suggests that we're just trying to we're capturing noise in the training set, however we decide to construct that, which, again, tells you that there's not a lot of signal present in the data that we can utilize to accurately assign individuals back to the geographic area in which they were sampled from. So I guess in conclusion, we have improved the quality of our baseline data set by increasing and balancing the sample sizes for those sample collections. And the results presented here are very similar to those that have been presented previously and continue to support the concept that there's a single genetic group of Pacific halibut in IPHC convention waters. And this is supported both by unsupervised clustering analyses that fail to identify discrete genetic groups of Pacific halibut, and a limited ability to actually assign individuals back to the location. And yeah, so the next steps for this project are to begin to incorporate those summer samples into existing analyses to examine and compare patterns of population structure over a larger spatial scale, with the caveat that those are summer collected samples and may not be representative of spawning groups that are in those areas. Yeah, I hope that we can move on, or if there's any questions.
So, Andy, I think I've got the same question I had last year, which is when do you declare victory on this one?
Oh, well, I think, yeah, so this is right. Beating this one pretty good. Yeah. I think, yeah, well, now it's time, we'll shift gears and really put the cap on it, no more sequencing. We had, an opportunity to do a little bit more sequencing. And that was, again, to, I guess, just essentially try to make the data set a little bit more robust, hopefully that's kind of makes it easier to get something published. But yeah, this is as far as data generation is concerned, this is.
But if you had twice as many samples, you're not going to see population structure there, right?
No, no.
It's not there. It's-
Right, there's a lot there, yeah. It's just, yeah, it's just not there. Yeah. So.
So is that the next step then, or?
Well-
On to something else?
Yeah, well, as far, yeah, incorporating the summer samples, I guess that's maybe a little bit more exploratory, you know, and then maybe, you know, you could use that to, you know, justify, you know, additional work, but yeah, but yeah, it's really just, yeah, focus on wrapping it up, and then, you know, what's next, you know, we'll just have to come up with, you know, new question, right?
Sounds good. I mean, I'm excited for you to get to move on to what's.
Yeah,
next.
yeah,
Like,
right,
it
right,
Seems like, it seems like any more work on this.
Yeah.
is Gonna not be productive.
Certainly,
because there's just structure there, and you know, not to critique what you've done,
No.
because we didn't know that ahead of time.
Right.
But now I think pretty clearly we do.
Definitely.
So I guess my advice would be to move on to those new projects as quickly as possible.
For sure.
And I don't know how much that next step is just kind of tying a bow on this one.
Yeah.
Be able to be done with it.
Well, I mean, I kind of see personally, I sort of see, you know, the analysis of those baseline samples kind of stands alone. Because, you know, the caveats that go along with summer collected samples is it's just, yeah, kind of an exploratory type of thing, right?
So maybe should that really be their priority then, doing the summer samples if it's exploratory? And
Well, yeah, no, yeah, right. I guess not, you know, but you know, we have the data there, and it's, I want to say, you know, easy to just kind of weave it into, you know, the analyses you already conducted, but yeah, but I guess, yeah, certainly, yeah, maybe we should, yeah, really focus on, I guess, yeah, wrapping up the baseline analysis. Go from there. So
I think especially because there's some really interesting questions to engaging.
yeah,
Other directions.
Right.
That would be super productive and useful.
yeah, certainly.
Are there any other questions regarding this portion of the presentation? If not, before we move on to the next research area, there's one request that was in the report from last SRB meeting, and that was request 04 that stated a preliminary evaluation of the feasibility of using information on genetic differentiation of Pacific halibut parasites as a possible structure marker. So we looked into that, and there's actually a very limited literature on the topic. There's actually a couple of publications, one in Pacific halibut, published in 2003, and a previous one in Greenland turbot, published in the late 90s, using parasites as a way to establish any structure. Pacific halibut is a particularly difficult species because it's one of the groundfish species with the highest number of parasites. There was a publication previous to the one that described the use of parasites for assessing population structure, indicating there's 79 different taxa, not species, taxa present in Pacific halibut. So they went and conducted an exhaustive investigation on the types of parasites that were prevalent in Pacific halibut, and they narrowed the list down to I think it was 9 different parasites based on certain criteria. Those parasites would not reproduce within the host, that they wouldn't cause or that they would not be expected to cause a decrease in condition of that's it. And with those 9 parasites that were mostly nematodes, campsophthalans, they concluded that there was some ability to differentiate between northern and southern stocks, but not at the resolution any other technique. And obviously, we're seeing now that genetic techniques are much higher resolution in terms of distinguishing.
Sorry, you're saying that the genetics of halibut provide better resolution
Yeah.
than the genetics of the parasite?
There's a lot of caveats actually with regards to parasites. One is that the parasite load changes with age. That's been shown in Pacific Island. Obviously, all the parasites are, most of them actually are coming through the diet. There's dietary changes, ontogenetic. That means that the parasite loads are different between younger juveniles versus older adults. The difference between sexes. And that's most likely related to the fact that Pacific halibut females grow faster, larger males. So there's a few complicating factors, but the result of that literature search was that it's not a conclusive technique, and I think would require a huge amount of work to identify which parasites at which stage should we be looking at. So at this point, I think seeing what the results of the population genomics work provided, I think it's safe to conclude. And she just that really there's really no evidence for genetic differentiation with this stuff. But there is, that it was interesting to see that there was actually some work conducted in the late 90s, early 2000s. After 2005, really there's very few publications using parasites as a tool for determining population structure. But it was interesting to look into that.
Thanks for running that down.
So with that, let's move on to the next research area. That's where we have updates from Colin. So that's related to the histology-based maturity scale. So Colin, go ahead. You can change his slide.
Great. Yeah. So got a lot of exciting updates for the review board. So we have 3 years of histology data now. I've broken this down presentation into 2 sections. One is the modeling of the histology data from 2022 to 2024 and the creation of a new coast-wide curve with those 3 years of data. And then we're going to move on to the visual-based maturity estimates and how we're using the new histology information to create a calibration factor to correct the maturity, to create a time series that's more accurate with the new histology information. So as you recall, we've gone out on our fishery independent set line survey the last 3 years and collected maturity samples. We've been able to collect over 3,200 samples over the last 3 years, which is an incredible sample size for this type of research project. In 2022, we were able to collect across all 4 biological regions. In 2023, the FIS was reduced down to biological regions 2 and 3. And then last year, we were able to once again collect in regions 2 and 3, but also get samples back out in the Bering Sea. So we have 2 years of data in region 4, but unfortunately only 1 year of data in region 4B. And I'll explain a little bit. The survey this year will be going back out to 4B, so we'll be getting samples back out there again. So as you recall, from SRB025, we came to an agreement about using the GAM model moving forward. It was a better fitting model than the GLM. So I reran the GAM with region and year effect in the model for all 3 years of data. And first I'll present each year sequentially so you can see differences. And then I have a plot that shows all 3 years combined by region. So here you'll see these plots a lot through this presentation. You have all 4 biological regions presented, and on the x-axis you have age by year, and on the y-axis you have proportion mature. And then those individual dotted lines are age at 5%, 50%, and 95% maturity by region, and it kind of gives you a better visual representation on how the different regions are different. So you can see here in 2022, which is a common trend throughout the years, you see that region 2 in particular compared to region 3 is showing a dip in the proportion mature roughly between the ages of 10 and 20. And as you can see in region 3, that there's definitely a steeper increase in proportion mature at younger ages, indicative of the A50 value, which is shown there by a little bit less than 10 years old. And then region 4 and 4B are also showing a steeper incline compared to region 2, with especially 4B is almost very similar to region 3, a little bit more variance because the sample size is smaller. And region 4 is showing A50 of about 13 to 14 years old. Then if you look at 2023 with just region 2 and region 3, once again, you see region 3 is showing a steeper increase in the proportion mature compared to region 2. And once again, between the ages of about 10, to about 17, 18, region 2 tends to have this little dip in the maturity ogive, which showing a lower proportion of mature females at that age range. And then if you look at 2024, once again, region 3 is showing a much steeper ogive compared to region 2. And region 4 is very similar. If you look at 2022 and 2024, region 4 is very similar in its thing. But one thing that you might not be able to identify that you can see now in this plot with all years combined, you see that over those 3 years, there has been a shift to the left. If you look at regions 2, 3, and 4, where we have multiple years of data, you can see that there definitely is some significant temporal trends indicating that the maturity has changed through time. And in region 2, you can see that 2022 was much different compared to 23 and 24, which were very similar. But then in region 3, there was a small incremental step to the left and higher proportion mature at younger ages through those 3 years. And then that same trend holds true in region 4. And unfortunately, we only have that 1 year in 4B. So we don't know if that trend is continuing in all 4 regions, but my suggestion is it's probably true across all coastwide. So now that we've looked at regional differences through time, we wanted to create a new coastwide curve with all 3 years of histology data. So we removed the year effect of the model and combined all years by region. And these are the model outputs for each region from 22 to 24. And then ultimately, we wanted to create that new coastwide curve. So last year, we showed our methodology by using weighting of the regional abundance estimates from Ray's FIS space-time model. And we used that again. But now that we have multiple years of data, what we did was we took the average of the index for all 3 years to weight these regional curves. And that gives you this solid black line indicated on the figure on the right. And it roughly falls between regions 2 and 3, which is what we would expect because those 2 regions currently have the highest abundance estimates from the FIS index. So this new coastwide curve is giving us a new A50 value of 9.8 years old. Now, if you compare that to the current maturity ogive in the assessment, that curve gives us an A50 of 11.6. So you also have to remember that these aren't apples to apples comparisons. So the current assessment ogive that Ian has been using is visual maturity estimates, not histology. And it's only taken in years 2002 and 2003 from regulatory areas 2B and 3A. Whereas the black line is incorporating histology estimates coastwide across all regulatory areas and biological regions. So this definitely shows you that there has potentially been a shift to the left in the curve over time. So one thing that we can look at to potentially infer into why there's regional differences is to look at the actual reproductive development of the females through the calendar time of sampling. So the FIS, runs through about May through September. So we have histology samples from those months. And you can see here on the left, you have region 2, and you go across through all 4 regions. These charts are all the data pulled together across all 3 years to look at potential trends across region. And what you see is that there definitely is some differences in how the females are developing through the calendar year reproductively. So you can see, I kind of consider region 3 kind of the poster child, because that's where we originally did the reproductive assessment project back in 2017 and 2018 to look at the full calendar year of female development. And what we found histologically is similar in region 3 compared to what we found in Port Locke region, which is the Central Gulf of Alaska. And that is that you see a large proportion of females that are already mature by May and June. And as the summer months progress, you can see that they are slowly progressing through the vitellogenesis stage and into late stage vitellogenesis by August and September. Now, if you compare region 3 to region 2, you see that there's definitely a delay in female development. You see that there's a larger amount of individuals that are immature in the earlier summer months. But as they progress, there's a slight delay compared to like July and August, what you see in region 3 compared to region 2. So the females in region 2 are slower to develop in their reproductive cycle. And then in particular in region 4 and 4B, you see a large, especially region 4, you see a very high amount of mature females. But if you just look at the females that are mature, which is the warmer colors on the top part of the bar, you see that as you get to the middle and towards like July and August, there's a higher proportion of those mature females have already progressed to a later stage in development, in particular compared to like region 2 and region 3. And you definitely see that in region 4B, where we've only collected samples in June, but about 50% of those females that are classified as mature have already progressed to late stage or even completed vitellogenesis. And it'll be important for us to continue to monitor these trends, especially in region 4B and region 4, because if those trends continue, it could be indicative of those females potentially spawning sooner than we thought they might be. Because if they're already reaching late stage vitellogenesis by the middle of summer, there's potential that they could be spawning in the fall earlier than potentially the winter months that we thought was happening coastwide. So this kind of is a good way to visually represent why there might be differences in the maturity ogives by region. And then just in conclusion for the histology section of this presentation, I just wanted to put up a figure here. So we are continuing to collect histology samples on the FIS. The vessels are out on the water right now. We're going to be doing both maturity and fecundity samples once again this summer. The FIS is reduced to lower levels once again this year, but from a biological standpoint, it's still great for us because we are going to be able to collect samples across all 4 biological regions, which we haven't been able to do since 2022. So now I'm going to move on to the visual maturity estimates. So one thing that we started thinking about once we went through the histology modeling was how do the visual estimates compare to the histology? So every fish that got a histology sample from 22 to 24 was also visually represented maturity or immature classification in the field on the setline survey. So these are the new regional models using the visual estimates, using the same GAM model that we use from the histology data. And once again, you can see that the trends are very similar. Region 3 is showing a slightly earlier A50 value compared to region 2. There does tend to be a little bit of a dip in region 2 compared to region 3. And the region 4 and 4B curves are pretty similar to the histology curves. So using these regional curves, once again, we created a coast-wide estimate using the visual estimates, using the same weighting method of the index from the FIS, the average index from the FIS spacetime model. And that gave us this new coast-wide curve. So now, if we look at this coast-wide curve of the visual estimates, it gives us an A50 value of 10.3. Now, if you look at the comparison across all three estimates that we have now, you see that the current assessment ogive from 2002 and 2003 gave us an A50 of 11.6. Our new visual estimates from 22 to 24 give us an A50 value of 10.3. And our histology gives us an A50 value of 9.8. Now, once again, just remember that the blue and red aren't exact comparisons because the blue line is taking samples across all 4 biological regions and coast-wide representation, whereas the current assessment model was only in regions 2 and region 3. So this gave us an indication that there was a potential shift even in the visual maturity estimates from 2002 and 2003 to now in 22 and 24. So ultimately what we wanted to do was we wanted to create a time series that was consistent with our more accurate histological assessments in order to input it into the stock assessment. And so in order to do that, what we did was we created a calibration factor between our visual and histology estimates from 2022 to 2024. And so to do this, we created a calibration factor at age, which was estimated between the difference between the histology and the visual at age on the Loggett scale. And the reason why the Loggett scale was used was because it ensured that our probabilities remain between 0 and 1 for the resultant calibrating curves, which I'll show you. And so because we in our previous modeling, we basically created maturity ogives by region and then weighted it to create a coast-wide curve. We wanted to also create calibration factors by region, which is what's represented here on the figure on the right. And what you see is that if it's a positive number going up, that means that it's a shift in any given age up. Or if it's a negative number, it means it's a shift down in maturity at that given age. So basically, if you wanted to put this in perspective on these regional calibrations, if it was a straight line across at 0, that means that every fish that had a histology and a visual estimate in 22 to 24, every fish was classified correctly for the histology and the visual. And that would basically be a calibration of 0. So now that we have a calibration factor, what we did was that we first fitted visual maturity ogives by year using a 3-year rolling data window from 2002 to 2024. And the reason why we used the 3-year rolling data window was because it allowed us to make sure that every biological region was represented during that 3-year time frame. If we were to use only 1 year or 2 year, as the FIS has been reduced over the more recent years, some of the biological regions wouldn't have been captured in that time window. And it also was comparable to our current 3-year estimate with the histology data. And then we also went back to 2002 in our time series because that was when we changed our aging estimates from visual surface aging to break and burn aging method. And so that's why we went back to 2002. And so you can see here with these visual curves, it can be a little bit hard to see some of the trends, but the darker colors are the ones that are earlier in the time series, and you get to more warmer and very lighter colors as you get to more recent years. As you can see here, starting at about 2002, 2003, 2004, it's kind of like right in the middle of the time series. And as you've gone through the early 2000s through the 2010s, the curve actually shifted to the right. But now in more recent years, the maturity has actually shifted to the left. So there definitely has been a lot of temporal changes through time. And it's been kind of nice to be able to visually represent this. We haven't done that before. So then what we did was we incorporated the calibration factor to create new calibrated visual ogives, and that's what's represented here on the right. So then now that we have new calibrated visual ogives, in order to create an accurate maturity ogive that we wanted to put in the stock assessment, we averaged across all three rolling data windows to create a final coastwide maturity ogive. So basically that was just taking 3-year data windows like 2003 to 2005, which is and then averaging 2004 to 2006 and so on through the time series. And this figure on the right is a representation of what the average mean fitted visual curve versus the mean calibrated visual curve is. And you can see that how the calibration has changed that time series. So ultimately, the solid black line is our new average calibrated visual curve that we ultimately are going to be putting, that Ian will describe in his stock assessment presentation. So now you can see a comparison to what our new visual calibrated visual maturity ogive looks like compared to the current assessment ogive. And we have 3 representations here. One is the calibrated and one is the truncated of that curve. And I'll explain what I mean by that here at the end. So our new calibrated curve gives us an A50 of 11. And our current assessment is 11.6. So there has been a little bit of a shift to the left through our 23-year data time series. But ultimately at the end, so if you remember in the current maturity ogive in the stock assessment, the maturity was truncated at age 8 because that was what our representation was of no females being mature before the age of 8. But now histology, we have seen that there has been 7-year-olds that have become mature. So now we have truncated our new calibrated curve at age 7 because the GAM model of the calibrated curve did provide some small proportion of females that were mature at ages 5 and 6. But histologically, through 3 years of data, we have never seen any 5 or 6-year-old. So as of right now, we're confident in saying that there's no females that are mature less than age 7. And so that's why that's been truncated. And Ian will go into the sensitivity analysis of those 2 truncated and calibrated curves in his presentation. So just in conclusion, first going over the histology-based maturity estimates. Once again, through the 3-year data time series, we see that region 3 in particular continues to show a higher proportion of mature females at younger ages. And the regional and coastwide ogives have shifted slightly to the left through that 3-year data time series. Now looking at the visual-based maturity estimates, we have seen some definite temporal changes. As you saw that curve in the visual, it shifted to the right, and now it's shifting back to the left. It'll be interested to see if we can figure out maybe what is causing that, whether that's environmental factors or different things going on coastwide that might be shifting those maturity estimates coastwide. And then once again, the calibrated coastwide ogive has shifted slightly to the left compared to the current assessment ogive that Ian's been using. So what's the next steps after this? The next step in our reproductive journey is fecundity. So since 2023 and 2024, and once again this year, we have been collecting fecundity samples on our fishery independent setline survey. And ultimately, the biggest question right now for us to answer is female Pacific halibut fecundity proportional to body weight. Because right now we are assuming that it is an equal proportion in the assessment, but what we don't know is if female Pacific halibut exhibit potentially a hyperallometry as they get really big, are they creating more eggs? That's something we just don't know in females. And so starting this summer, we're going to be starting to go through the fecundity samples and hopefully make some progress on that and be able to present some new information to you guys next year regarding fecundity. And I think that's the end of my, I'll take any questions that you guys got.
Go back to slide 12. So what are the point sizes here are the number of individuals?
Yeah, I'm glad you brought that up. That's one thing I noticed. We didn't put a representation. Yeah, the individual dots are sample size.
And what does that mean? Is the dot the average of those individual, like?
No, it's just
what is the sample?
the dots are more of just a visual representation of the sample sizes based on age.
So
Number of fish per dot basically is that.
So the number of fish in a dot can come from different physical samples.
It's basically the raw data visually represented by dots. That's what it is. That's what it is.
But the model was fitted.
But the model is fitted by the raw data.
To individuals.
I think it's been, if I remember correctly, it's been the dots are bin data by age. So that way you don't have, so that way it's, otherwise the plot could get a little messy because you would just have like individual dots by age.
My question is, so the individuals that make up, say, a dot at age 10 can come from very different locations within region 2.
That's correct.
I'm trying to think of it, if there's implications for kind of pseudo replication.
Yeah, we talked about this last year, Lou.
So,
Well, we potentially think that could get resolved using more spatial modeling, and that Ray's not here. I don't know if Ray's here today. That is something that Ray has started looking at because Ray he has begun to create a coastwide. I don't know if Ian has more. He's created a coastwide mesh, right, for his.
Yeah, he's been working on a geostatistical.
Yeah, and he's actually using the maturity data as kind of a test run. And so that's kind of in the works, but it was something that we did talk about last year. Yeah. If you do go through the literature and everybody that's doing maturity estimates, especially with histology, they're basically, the issue with histology is that you take the samples as you can get them. And I do understand your concern about the pseudo replication side of things. But this is basically how things are done on creating the maturity ogives, because it can be very hard to get these kinds of samples. But in-house, we are blessed to be able to potentially look at more spatial modeling to maybe offset that type of replication.
so the visual, sorry. Well,
Go to the visual as well.
oh, okay. I just wanted to bring up visual showed a tail off, the declining proportion.
Yeah, so
And that raised issues of skip spawning and going down that road. Is that something that is probably not going to be as prevalent now, or are you still going to look at that?
we have had a lot of discussions regarding skip spawning. And there's basically two ways that could be incorporated into the assessment. One is you look at mass atresia and you indicate that a specific female is not going to spawn for that any given year. Now, a lot of people have decided to use that in their maturity estimate. We think it might be a better way to incorporate it into the fecundity side of the total picture of maturity for female Pacific halibut. Because when you just look at the maturity estimates, that is basically just, is the female mature or not mature, right? It's an immature or it's a mature female. Whereas if you incorporate atresia doing that, it might be an incorrect representation. But if you incorporate the atresia to potential reduced spawning of egg output, we ultimately think that might be a better indicator of how skip spawning or mass atresia could be incorporated into the maturity estimate.
But it can go either way.
It can go either way, right?
Yeah.
Yeah, because there's also been a lot of work on the fecundity side that's looked at down regulation. So there has been some species of fish that will actually, as they get closer to the spawning event, they'll actually have atresia or reabsorb some of those oocytes before spewing them and broadcast spawning into the water column. And that side of it is another aspect of the atresia, mass atresia, and looking at seeing whether we can see in female Pacific halibut as they move through the reproductive cycle, is atresia getting more and more and more as you get later in the year? Or is it just like this female knows in June, I'm done, that type of thing? And that's something that we still need to figure.
I guess the first question is whether that decline is real. I mean,
Yeah, so if you go,
there's still a few samples out there.
there actually, that is one, Ray and I ultimately decided to just go ahead and fit the data. That drop in region 3 is actually just a couple of fish that were indicated at age, I think 25 as immature. And I think there's really like no data past that. So that is something that we could talk.
That's what I'm talking about.
Yeah. So that could be something that we can discuss. We ultimately, I think we just wanted to present what the model showed. And then, but ultimately, if that's something that wanted to be correct to get put into the assessment, or Ian maybe might have an input on how much of an impact that's actually making on the assessment.
There's so few fish out there.
Or even at that age, right? So that is something to think about.
So did you do a GAM that has method in there to determine whether method is actually?
I don't believe so.
I think that would be a good first step. Okay. Before going down the road of splitting everything out by method, visual or histological. Just to establish that there really is a difference in maturity estimation.
Okay.
May also be worth noting that there obviously could be a biological explanation for higher ages, but there also could be misclassification in the field. So that's something that we're going to be fine tuning the macroscopic classification with the histological. There could be a misclassification of immature females when they're actually mature.
I mean, this brings up a larger question I had about how you come up with the coast-wide estimate. And what I understand now is you do a weighted average for each age, each individual age band. And that's how you come up with it. Is that right?
Yeah, I'd have to think about that for a second. I think it's just waiting. Yeah, I don't know if it's just weighting the entire curve or it's weighting by the individual age. Ian, do you know?
I believe we're fitting independent curves to each region.
Yeah,
The
and then
issue-
weighting the curves.
The issue is that we sample regions at different proportions, so we can't just then take a naive average.
I think there's not a really simple solution here. But when you do it that way, you don't account for the uncertainty different regions. I mean, an alternative would be to do a hierarchical of regions nested within the coastline. That would that would help you account for some of this balance and sample sizes that you dealt with by this time. It wouldn't allow you, at least in a standard sense, to weight by the relative abundance of the different regions, but you might be able to do that coastline somehow. I think you've solved some problems by doing it this way, but
Yeah.
but there are other problems that are not being solved that way, and it really comes out here where when you weight by region, that region 3 dip shows up, and it really shouldn't be showing up if you took into account the uncertainty, the confidence intervals there. My intuition here is that that's not real. It's the result of 2 fish. Somebody squinted at them, put down the wrong answer. Those 2 fish. And with that confidence interval like that, some approaches, like a hierarchical approach, would say, we're really going to downweight this because of the high uncertainty there, and it's not going to affect the coastline range. But because of the way you did it here, because you wanted to solve the different issue.
Yeah.
it
Yeah.
results in that being weighted just as highly as else.
Although if you go to the curve, the one where you show all the visual for all the years. Sorry, each year's visual estimate.
The color,
Yeah, that one.
this one is the.
That's the calibrated.
That's the calibrated.
Sorry, the non-calibrated, so. if we're
That's the-
just based on visual.
This is just the fitted models. Go one. Yeah.
That one. You can see that decline is pretty,
Gets rid of it here.
yeah, and that's because it's really most pronounced in that one sample.
It's because this is using a 3-year data window, whereas the previous was only using that 1 year, right? Yeah, so that's why this looks better than that, because it's a 3-year data.
But you're totally right. We focused on weighting issue because we're worried about this sampling.
Yeah.
Area.
Yeah.
And the fact that the abundance itself isn't uniform.
Well, I think that's the main one, right? I mean, you don't want to have regions with very few fish.
Given equal weight in the average. Right? But you're right, it doesn't necessarily capture the certainty within region when you combine across the regions.
That's why, again, the space-time model of maturity is really the way to say it. It sounds like.
Where's Ray? We need.
That sounds to me like
Yeah.
overkill, but if he's already going down
Yeah.
that route already to deal with the spatial issue, you might as well do the whole analysis right
Yeah.
and deal with all of those issues together.
Well, we-
So I think if we talk to Tim about that and see like whether that's a step beyond what he's hoping to do here.
Yeah.
But this analysis is not exactly correct because you deal with one issue or you deal with the other.
Yeah.
And I don't understand, I don't have any good suggestions for how to make this formally correct other than the whole space-time model.
Yeah.
Other issues on maturity?
I just want to make sure we are going to talk about this with Ray at some point. Maybe tomorrow, yeah.
Yeah.
What a good time to break. Yeah, we're ahead of time. So
Well, yeah, I don't know. Schedule seems to be somewhat irrelevant. What's happening?
I will likely help us fill the rest of the morning.
Yeah,
Before we start
The assessment's gonna be
Quite a lot.
a lot.
We actually only have a few slides, so we may just want to finish now and.
Yeah,
then
let's
take a
finish
break.
at 10, aim to finish at 10:30. No, take a break till.
Yeah, the next research area is mortality and survival assessment, and here we have completed the 2 main activities that were contemplated within the current program of research and monitoring. One was the discard mortality rate estimation in the black fishery that's completed and published. Then the second one, the discard mortality rate estimation in the charter recreational fishery, that project has also been completed, and we're in the process of writing the manuscript. So that brings us to the very last.
Can I just interrupt for a second?
Yes.
So what is the estimated discard mortality rate in the charter sector?
That's an average of 1.35%.
Precise, 1.35%.
With the best,
Huh?
with the best handle. Really good condition, really.
Fish in excellent condition, but it was hard to make them down the index.
So you think it's good representation of the fishery?
It doesn't cover all aspects, and you'll see YouTube videos of and fish less than that. But this is the focused on the charter recreational fishery where you have a guide, and I'd say it's quite representative.
And I assume this is probably a big deal in 2C. That we're,
It's a big deal, probably in 2B, 2C, and 3A, and we do the study in 2C.
and how big is that recreational catch relative to the commercial? So like what kind of total mortality?
So the rec sector is typically in the 15% of the allowable catch range. That's a very small amount of discard mortality that's happening in that. Now, how much of that 15% is charter versus weekend warrior or unguided? I don't know that off. Yeah.
I asked because my brother-in-law flew from Boston out to Lodge outside of Sitka somewhere. And, you know, he was describing to me all the fish they were getting and processing. And I was like, that's a commercial operation. And just trying to get us an idea of like the scale of the release mortality. Because they went through a lot of small fish. Okay.
So closing in on the last research area that we're going to be providing just very brief updates is fishing technology, and that's in relation to the project that we described previously in this regards to investigating new methods for whale avoidance and deterrence to reduce whale depredation in the longline fishery. So I'm going to pass the word to Claude, and he can give brief
You can keep
summary on the progress in this.
Yeah, so we just got back from our field work on that. Myself, Ian, and Josette were out on the fishing vessel, the Oracle, for 8 days out in the Aleutian Islands, Bering Sea area, which got back last weekend. And we had successful deployment of the shuttle. This work focused on the shuttle gear. So this is a device that kind of zip lines down to the ocean floor on the ground line, removes the fish from the hooks at the bottom of the ocean, and then brings them up in a protected kind of steel container. We were looking at refinements, the ability to attach it, how long does it take to attach it, how does it perform, both in gear that's unprotected versus gear that's protected, both in the presence and in the absence of whales being around. We were able to collect data on that. We've got video footage from inside the device to understand how many fish are coming in, how they're being released, how that may affect damage to the fish. We also had cameras on the outside of the device to capture any fish that were being excluded from entering the device, because sometimes the weighted gear comes in, or if the fish is big enough, we're concerned they may not get entrained within the device. That all has to be turned into data. We also had cameras on the control gear, which didn't have the shuttle come down it, just to see if we would see depredation in action. And I think we got really good catch comparison results. Some of the presence of whales is going to be more challenging. It seems like Murphy's Law is probably going to raise its head on some of those sets, but maybe not all of them. A good example of that is on the control gear. So all the gear was on one string. But on the control gear, we had 20 halibut lips come up while we had whales around. So then we went to set the shuttle down, and the shuttle came up. It had rockfish in it. It was like that section of gear, which was further along the line, had moved into different habitat possibly. And so again, we have to review the video footage to better understand where they're halibut in that area, where they're not, things like that. But overall, I think it was a success. We got chased by the weather a lot, but we got some good solid data to push through now. It was
Shuttle device hit the bottom, it goes on the bottom, right?
It goes to
Goes and rests
near
on
the
the bottom?
bottom. So sometimes, based on the video footage, it's often just above the bottom. So as the ground line is lifting up, it is sliding forward. Sometimes you see the leading edge of the collar of the shuttle kind of dragging in the mud a little bit, but most times it's off the bottom. It's not. So the fish are being lifted maybe a foot or two into the water column and then into the device. And that generally worked. So occasionally we'd hit on its way down, we would encounter a snarl, which would then temporarily stop the device, lift all the gear up temporarily until it and they would typically
Pass through now?
it can pass through. So it has this kind of keyhole device that it's generally sliding through it, and the snarl will actually pass through a circular space. It'll tighten up and pass through a space that big. But yeah, so it was interesting when the whales were present, there'd be a cluster of them, say over at that next building where they were really not near your boat. You could see them. And it was hard to tell if they were actively diving on the gear or whether you're seeing 80% of them currently breathing and some of them are actively down on the gear. And then you'd have two that would come right up next to us, like within feet of us.
Is it sperm whales?
They're killer whales. And it was just an interesting dynamic. It's like, what's going on there? And are there individuals right now actually on the gear? And the way you'd find that out is typically you get halibut lips back or sailfish back. But we would frequently have them just charge at the boat. You're on a 58-foot steel boat and you're like, what are you doing? Like they're coming at you and you're like, you're feeling kind of safe, but you're not 100% sure what they're doing. And then they'd roll onto their side about 6 feet below the vessel and blast it with air bubbles. Wow. It's like, what are they doing? We're not sure if they're doing that. Like there was a pot here going on in the area for black cod. We're not sure if they're going underneath the pot here and blasting air at it to scare individuals back out of the pot or into the escape rings and then grabbing them that way. And if they're testing near the vessel to see what exactly the vessel is or what that behavior is. But it was interesting to see that. We didn't, to my knowledge, and there's hours and hours of footage. I don't think we captured any of that on video camera, which isn't unusual. Often the whales are pretty cagey to new devices around them, and they kind of keep their distance, especially at first, until they get comfortable with what this is and start investigating it. So we hope to capture individual video of that, but it's not.
So it seems like they're figuring out the spot here.
We're getting more and more reports that they're what we call treating them as pinatas. So they go up and they tear them open and then they get, 150, 200 pounds of fish all in one go instead of. And there's evidence now that they're doing it in the true pot fishery, which is a rigid pot that's being used for sablefish. And we were hearing reports that one guy had 70% of his pots torn open on that trip. And there's
Still
big,
just nylon mesh.
right. So it's big congregations of whales now. Like they're in the 20 to 30 individuals around the vessel. Like it's not just 3 or 4 individuals, but it's big groups. So you could very well have 12 animals at the surface, and there's like 6 to 10 down below doing something that you're not seeing.
We actually called that vessel and had them come drop the whales off on us. They were headed in after fun fishing. We set a string of gear. We called them. They drove over to us. The whales peeled off onto our gear. And when we started hauling here, we had 30 whales working there. So they,
You wanted them there.
well, we were, yeah, we wanted some replications with whale activity. There was an effective handoff there, probably the only one of those ever that's desirable.
They're, you hear that they can be efficient, although it's really hard to interpret, right? If your gear just come up because it got cleaned off by some vertebrate on the bottom or what happened to it. But it seemed when the whales were there, they were really efficient clean gear. Like I was a bit shocked by that. It was like, there was very little evidence that anything was like, it was almost like a water haul. Except for halibut seems to leave their lips behind and sailfish don't,
Well, that's a good indicator.
right? But I don't think, there's probably halibut coming off. They're not leaving their lips behind. And sablefish aren't leaving much evidence behind on them. They are biting right off. I suspect they're tugging them off a lot. Because if you look at control gear before they show, they're there. They're in that area. Those like are large quantities of sablefish. But then you'll get one sablefish head and hundreds of them. So.
Hopefully in September, we'll be providing some of that data, maybe some of the footage as well.
Right.
That'll be interesting to see. So, well, this brings to the end of our presentation.
Okay, thank you. So we were scheduled to end this session at 12:30.
Yeah, it's the old provisional agenda. So we normally have a break from 10:30 to 11.
Do we need 10:30 to 11 or? Yeah. Well, let's just go at 11. Come back at 11, 20 minutes.
So just a reminder, we're up to presentation 07, please.
Yeah. Excellent. Okay. Sure. Yeah, hopefully that's all right. Go ahead and just use it from there. And I can't see the chat, so if anybody who's on, if there are questions in the chat, just. Okay, so I'm going to provide an outline to begin with, following generally the format that I have for previous full stock assessments. There's a lot of material here. You probably unfortunately noticed as you went through the background materials. It's a pretty substantial document. There are a series of other related files and things, depending on how deeply you wanted to go into it. There's basically all the detail for all the models, output, supporting papers, a whole lot of things. Obviously, I don't have time to present all of that information today. So the way I organize this presentation is to really provide essentially highlights of each of the sections and give us an opportunity to have questions and discussion about any of the various topics as we go through. And I'll try to highlight some of the things that I think are most important to consider and perhaps some places where there are questions, open-ended questions. You'll also notice that we have in previous SRB meetings, we've had the biology section after the stock assessment, but we felt it was important this year to have it first because the maturity curve is one of the things that I've evaluated in this year's stock assessment. In fact, that actually might be a nice format going forward, just to have the biology products are there before we start talking about assessment and parks policy. So in this outline, I'm going to give you a quick refresher on the IPHC process. I'll touch on recent SRB requests from the last year. I'll go through some highlights of the data sets that we have available to us for the stock assessment and some of the MSE and harvest policy related items. I'll look in a little bit more detail at some of the new and revised information that goes into this year's stock assessment, highlighting places where we've made any changes to the underlying data sets. And then continuing on, I'll direct you on the multi-model approach that we take to the stock assessment, outline the structural assumptions, and then I'll go through the bridging analysis where I outline each of the changes from last year's assessment to the preliminary assessment for this year. We'll look at a few diagnostics and results. I'll talk about quite a bit about evaluation of uncertainty. And as you'll see, I've got a wide range of different sensitivity analyses, as well as retrospective analyses. I'll talk a little bit about the preliminary results for 2025, noting that these, obviously, there's a year of data before we get to the final assessment. And then we can finish out with a brief recap of updated research priorities and avenues for future development. So that's my proposed outline for it. And you'll notice that I've got a few photos in here of some of the really classic buses, including the Polaris, who's going to be working on the survey for us. This boat was built either in the 19-teens or the 1920s, still fishing. It's in its fourth generation in the same family. And we chartered them to work on our survey this year. So pretty cool history there. Okay, so the IPHC assessment review process. You all are fairly familiar with this. Our general approach is that we are doing annual stock assessments, but we do full stock assessments approximately every 3 years. The last 3 have been exactly on that schedule in 2019, 2022, and then this year, 2025. In between, we do updates, meaning we include all the new available data. We also have, in some cases, added new pieces or made change to the model, but generally we're trying to limit major changes in the update cycle and save that for the full analysis. I'll highlight a few times in this presentation some of the changes that have been made on the 3-year cycle and some of the proposals for this year. I think generally the participants in the IPHC process appreciate that we're not trying to make major structural changes every year. A little bit of continuity is a good thing, but it is important to keep things sharp, to keep refreshing and not get behind. And looking back through the history of our stock assessments back into the 1970s, the times when our assessment has had trouble is when we've not had at least a 3-year update of all the basic assumptions and making sure that it's as current as we can. So the annual cycle within that 3-year cycle, as you know, is that we look at science and research in the June meeting, and that gives a sort of a prospectus on the year. Then we have the summer to address any comments, questions, improvements, and we then refine those and finalize them in September. And then after September, things are pretty locked down. So it's such a tight time frame after the September SRB meeting. Really just adding the current year's data, turning the crank, getting the results, and then meeting the management. That's the meeting and management process. So the results are first presented at the interim meeting, which generally falls around Thanksgiving, and that provides the public and everybody else an update of the process. And then the results, essentially unchanged, are presented again at the annual meeting in late January. So everybody has about a month to a month and a half to digest the results, to ask questions. We do a series of outreach presentations to different groups to make sure everybody has the information they need and the real decision making at the annual meeting in June. So for this stock assessment, you'll note number 7, which was published back in early May, along with this presentation. That provides essentially the outline overview of the information. You should have received a link to our SharePoint file, which provided the detailed model files for each model, graphics, outputs, the technical documentation for the stock synthesis software. Also included there last year's full stock assessment documents in case you wanted to look back at the documents that were provided for the commissioners. And a few relevant manuscripts just to save you the trouble in case you want to track something down. You couldn't include all of them there. And then on our website, we actually have a compendium of all of the quantitative stock assessments back to the late 1970s. Prior to that, we were generally doing CPUE-based sort of equilibrium methods that weren't really stock assessments, they were more sort of management approaches. So really, from the start of quantitative stock assessment at the commission, we have all those documents available, and they're all linked on the website. So somebody could go back through and read. It's close to 50 years of stock assessment now if they wanted to. Kind of neat to have a lot of history there. So I'll go then. So that's the process. That's just a general overview of how these work. So this is the third year, so this is stock assessment. And I wanted to recap the 3 requests that came out of SRB 25 last fall. They're all here. I'll go through these one at a time. So the first one was a recommendation to consider the impact of FIS designs, not only on the abundance, but also on stock distribution across regions. To refresh you, we create an abundance index or indices that from the geostatistical model that we produce every year. And those provide the index of numbers per unit effort in the stock assessment. They also produce an index of weight per unit effort, of legal size, and all sizes of fish. And those are used in various aspects of decision making. An important one of those is that we also get an estimate of the stock distribution from our survey, and that feeds into the decision making process. And that's a place where I think we have not, until last year, we had not extensively evaluated the role of uncertainty in those estimates. Essentially, we would get a vector of percentages by area. The commission would have those at their fingertips when they were making decisions, but they weren't explicitly doing anything with the uncertainty associated with those stock distribution estimates. And in the last couple of years, as we've had a fairly sparse design, and this is a figure that Ray created that I think really well captures, I think at the request of the SRB, really well captures. Right. Oh, the RAB, right? Our Research Advisory Board recommended this. This really captures how recent our information is. So you can see that the lighter colors represent more recent sampling. So it shows you, well, prior to this year's survey, how recently have we sampled each of these different areas. So the darker the colors, the longer it's been. And you'll notice a lot of that purple out west, it's been 4 or 5 years plus since we've sampled some of those areas. We're starting to generate a lot of uncertainty, and particularly 4B. You'll see there's no colors out there within the last 3 years now that we're in 2025. So we wanted to find a way to capture that. So the way we addressed this at this annual meeting 101, which was January of 2025, was in black would have been the standard outputs that we would provide for the commission during their decision-making process. So the legal size or O32 stock distribution, that's the percentage estimated in each of the regulatory areas. And then that gets turned into, based on a weighted exploitation rate approach, that gets turned into a set of TCEYs. Now, the commission obviously doesn't have to use these TCEYs, but that gives them sometimes a starting place for discussions. We added this year was the range of uncertainty associated with our estimates of stock distribution. Now, obviously, these don't sum to 100% at the lower and higher confidence intervals. But what we wanted to show was, given the uncertainty in the survey, what's the range within each of these areas that of the stock distribution, and then translate that all the way into the TCEYs. And during the meeting, we really highlighted, for example, 4B, if you look at the TCEY down there in the bottom, given the uncertainty in the survey, the fact that we haven't done a survey out there in 3 years, the TCEY, just applying the deterministic approach, could be anywhere from a little over 1.5 million to almost 5 million pounds. That's a pretty broad range of uncertainty. I think this was really helpful in helping the commission understand the amount of uncertainty that was present in our survey data and translating that all the way through into the information available for their management decision making. So that's how we evaluated this and translated it through into management. Took some explaining, but I think it was helpful information for the commission.
Can I ask a question?
Sure.
So, I mean, that's awesome. I think that's okay to look at. What about the aspect of this that's alternative business? Looking forward, here's how this could blow up.
Yeah, and so I'm not going to get there, but Ray actually did quite a bit of work on that topic as well. Presented earlier in the process. I don't actually have any graphics associated with that.
I mean, this is bad, but
Yeah,
it could get much worse.
It could. and we yeah, we could dig up some of those figures. When we went through the design, so I guess the reason I didn't go there is because the design process was largely completed before we got to decision making. But during the design process, we definitely presented confidence intervals associated with credible intervals associated with different designs out to 3 years in the future, right? For different biological regions and regulatory areas.
I'd like to see those.
Yeah, we should get those. You actually, I think you may see some of that in Ray's presentation on upcoming designs. No, haven't done that yet.
Yeah, thinking back to our discussion last time, I think this came out of discussions about how the coast-wide CVs ask a lot of uncertainty, but
Yeah.
So showing that the coast-wide CVs were relatively robust.
Right.
They reduced this events, underestimate, so the impact of reduced this events.
Now, the challenge is that the use of this information is sort of part of the negotiation process that occurs at our annual meeting. And we don't always have very clear guidance about exactly what commissioners want to see in this regard. And so we have to be somewhat judicious about presenting on request as opposed to front-loading information that may or not be relevant to their.
Sure. I mean, you could eliminate the whole bottom part here about translating it to TCY and just look at how the uncertainties of the workings.
Yeah.
You wouldn't have CIs because you don't have years. It won't be a point estimate, CIs. We can have a CV, but we don't know about future in terms of frame distributions.
Well, sure.
So we have to talk about the same. The coefficient of diffusion.
Okay, the next request was with regard to the relationship between commercial CPUE and SWPUE. We touched on this earlier in the advisors' reports. For a couple of reasons, we haven't yet started on this request. The first is that we have a couple of updates or changes that are potentially coming through our database for commercial CPUE this year. And it seemed like we should try to get those in order before we did this analysis. And the second one was that because this was mainly a data analysis and not a stock assessment analysis, the focus this spring has been on getting the full stock assessment done. But this is certainly something that we can work on over the fall, over the summer for presentation this fall. Still definitely interested in this. And I think to paraphrase this request, my interpretation of this is looking for evidence of where the survey design has been limited. Is there any evidence of seeing different trends in the fishery than we would generate from the survey based only on the areas that we've been able to sample in these few years? There's some simple things we can do here, and then I think it could get quite a bit more sophisticated depending on how deep we go into this. So we've not tackled this one yet. So I want to leave this on the blogger in case we refresh it. And then the next one was with regard to research priorities and upcoming 5-year research plan. You'll see I've made some proposals for 2 or 3 items in each of the categories for the upcoming research plan, but also the suite of smaller topics that we maintain in the assessment, just so they don't get lost. So that I'll cover that at the end of this presentation, and it's also in the document. I might not be a presenter, but yeah, if I can get elevated again, I can make this slide. Thanks. Yep. got it.
While we're waiting, I'll note that up in the northeast US, most of the assessments are moving to that. So,
Yeah, and I think, as I understand it, they have added a what they're using is a multi-stock piece in it now, which could be used as a multi-sex. So that's been the big impediment to taking up WHAM, is that it doesn't have males and females. But I think the way they've implemented having two areas, it could be two sexes within an area if you find your data with that. So that's, as you'll see, that's one of my top research priorities, recommendations, is to move toward exploring a stage-based model using that framework for this, for halibut.
yeah, Black Sea bass. Two spatially.
It's interesting that they don't have any species that they think that dimorphic growth is important enough to need two-sex model.
Summer flounder has been one that the industry has pushed for a two-sex for a while. Some of Pat Sullivan's work showed there as much as we thought.
Well, nobody has dimorphic growth quite to the degree that we do, I don't think.
Yeah, so
We're,
some
we're,
of the flounders up
we're
there.
on the extreme of, you absolutely can't ignore dimorphic growth. But I believe that that's been now available, that new feature as of this year, 2025. So I'm excited to explore it. Okay, I'm going to move now into an overview of the various data sources, and I'll try to give you a little bit of a historical perspective, just so that you'll understand some of the challenges associated with modeling these data, and hopefully some of the background of why we essentially can't decide whether we need a long time series or a short time series in this assessment. Most places just decide on one or the other and live with it, where here we're actually using that as an axis of uncertainty. So Pierre Carpi led the effort just a couple of years ago now on this paper summarizing all of our basic understanding of life history. And of course, like everything else about Pacific halibut, the picture is pretty complicated. This is a conceptual diagram of what we understand to be the linkages between spawning grounds in purple, some putative spawning grounds, particularly on the west side of the stock that we don't necessarily have direct information on, nursery grounds in green, and some of the linking migration and movement, either at the egg and larval stage, the juvenile stage, or the adult stage. And in a nutshell, the stock is pretty highly connected. Not surprising given the results that we've seen from Andy. The stock is pretty highly connected across its whole geographic range. Fish can move over their lifetime. We have, as an example, we have fish tagged in the Bering Sea that were recovered in Oregon. So basically moving within their lifespan across the entire geographic range. And we have fish that were tagged in the summer and recovered 1,000 miles or more away in wintertime at spawning grounds, and then returning to almost exactly the same summer feeding grounds. So we have what's been called in some of the literature site fidelity, only it's kind of the opposite of site fidelity in most fish, is that it's not the spawning site fidelity, it's the feeding site fidelity. So it's like if you took the salmon life history model and turned it on its head, we have some fidelity to where they go in the feeding season, but not necessarily where they go in the spawning season, at least as evidence.
That same fish went to 2 spawning locations?
Well, we don't have direct evidence of that, but the genetics suggest that these spawning aggregations are discrete, that they're not just returning back to these spawning aggregations. That's why all of Andy's stuff focused on winter spawning samples, so that we could isolate spawning grounds as opposed to summer, when we assume that these fish are mixed.
Well, with the caveat that a whole lot of straying.
Yeah, and the tagging, we've put 500,000 tags out over the last 100 years. And we've got 50 or 60,000 of them back, and that suggests there's plenty of straying from small corners of the stock. I think in terms of the stock assessment, the biggest assumption we're making and imposing on this is that we're not explicitly modeling the Russian side of this. A few years ago, we were making some pretty good progress toward developing connections, getting access to data sets, and trying to think about developing a bigger model. And then with the events in the last few years, we now have zero ability to work with Russian scientists. So that's been kind of a disappointing dead end in recent years. Our understanding is that at least for juvenile and adult movement rates right now, we don't have strong evidence that there's a lot of fish moving back and forth from Russia to the US, but certainly some are. We have at least a few handful of tags that suggest that kind of movement over history. The density is relatively low on the very northern Bering Sea shelf, suggesting that even if there's a lot of exchange going on there, it's a pretty small fraction of the overall. But it is a recognized and currently unaddressed source of uncertainty in the stock assessment, which is that we would like to be able to assess the entire stock range rather than just the part in convention waters.
Wondering if there is very strong site fidelity. So actually then it would be interesting to sample, take the genetic samples and look at the genetic differentiation at the feeding grounds and the spawning grounds.
So that would be the summer samples.
Yeah.
And that's, those were we've done some of that, and that's some of what Andy was just talking about. Sure. Whether and how much analysis. I don't know, Andy, you want to comment on summer versus winter samples?
Yeah, I mean, I guess it gets down to like, yeah, there might, summer site fidelity might exist, but like genes are being exchanged and recombined on the spawning grounds, right? So I guess you wouldn't expect there to be like distinct genetic signatures associated with like the feeding grounds, right? So I think that's kind of where that, sort of breaks down. And like it works, like talking about site fidelity and like a species like salmon, right? They spawn once and die, right? So but you don't know with a species, that spawns many times throughout its life, like what is the degree of site fidelity? Like, do they return to the spawning grounds for every single spawning bout through their life cycle kind of thing? So I think that's, the difficulty with, trying that kind of a thing. And just, making sense of that and trying to interpret that would be difficult.
The other challenge is that it wouldn't, even in the cleanest case, it wouldn't be a lifetime site fidelity to those feeding areas. So for example, we have evidence from our pit tagging study in the early 2000s suggesting a high proportion of fish that were tagged at a given location were recovered very close to that location during summers. So there's definitely evidence of site fidelity there, but that occurred even in region 2 where we have almost no juvenile Pacific halibut. So those fish were born somewhere else. At some point in their life, they selected that feeding location and then apparently had occurred. I think we've got potential for mixing at various life stages that's going to make genetic protection. Anyway, this is all sort of background to illustrate that we don't think this is a fully instantaneously pan-mictic stock, but yet it's got enough mixing that dividing it up into isolated areas that don't account for all this movement doesn't work either. That's where we were in the early 2000s. We did stock assessments on individual chunks of this, and then when we did the tagging study, we found that the fish were moving across those boundaries, and what that was creating was a bias in those estimates. A bias high, because if you're estimating doing a stock assessment on a small piece of a larger stock and you have older or larger fish showing up, the only way for that model to explain that is to make the population bigger, because the effective fishing has to be self-contained when it's an isolated unit. And so there was evidence at the time that the sum of those individually assessed parts was larger than the answer you get if you assess the whole thing together. That was the rationale in 2006 for moving from a bunch of separated assessments to one combined assessment. Challenge there is that, of course, they're not instantaneously mixing. So now we have a little bit of stock structure inside a single stock assessment model. And yes, Sean would be the only one who can recall the 5 or 6 years we spent trying to develop a fully spatially explicit stock assessment model, which wasn't a failure. It didn't produce a management-oriented model that had spatially explicit dynamics, but it did feed heavily into the development of the operating model. And Alan took that initial work and has turned it into, I think, a really good operating model for us. So there's the potential someday for someone to pick that up and make another go at a fully spatially explicit.
I think that was from the beginning, it was like, this probably isn't gonna work, but we'll do it anyway. Just to,
Yeah.
because you knew the operating model was gonna need something like that anyway. But I don't think there was ever an expectation you were gonna stock assess.
There aren't many complicated spatially explicit stock assessment models that are directly used for management. And the reason is because the data overhead is just cryptic, technical. Even here.
You immediately get compounding between natural mortality rates.
And recruitment distribution. That was the big one we found. We couldn't delineate between fish born in one place at a higher proportion and then subsequently moving versus born everywhere and then moving to those places later on. A lag in our survey where we don't tend to see fish until they're 7 or 8 years old gives them plenty of time to redistribute from where they actually recruited to where we subsequently observed. So resolving that was difficult. And so instead of resolving it, Alan is able to actually just evaluate different hypotheses about recruitment distribution. In an operating model sense, it's a far easier task. Anyway, long-winded way of giving you the preface of why we're using a coast-wide stock assessment as opposed to individual areas or something complicated. We have just from first principles and from all of our evaluations of the data, we've settled on this concept of having a single stock across convention area. And the light gray is the convention area, noting that the darker gray in the upper left-hand corner there off Russia is still part of the species distribution. It's just not part of our convention nor this particular stock assessment. And within that, we've identified 4 biological regions that tend to have characteristics that are more similar within the region than among regions. Noting that there's definitely still movement across these regions, perhaps, definitely over the lifetime of these fish, and in some cases, probably even within a calendar year, there's certainly going to be some fish that move across those boundaries. But we found it useful to try and at least delineate into these 4 biological regions, which seem to have a much greater meaning than the individual IPHC regulatory areas, which really were designed for regulatory purposes and not based on biology, perhaps with the exception of the boundary between region 2 and region 3, which has a little bit more of a biological basis to it. So when we structure the stock assessment model, we have 2 approaches. One is to combine everything into coast-wide indices, and the other is to keep things separated by biological region and then fit those regions within the stock assessment model. To go back in history, this fishery developed in the late 1800s. It ramped up quite quickly in the early 1900s, and you can see that in the early part of the 1900s, we're already taking 50 to 60 million pounds out of this stock on an annual basis, and it was all coming out of region 2. So it was virtually all coming from the US West Coast, Canada, and up into parts of the Gulf of Alaska in that early time period. Then as the fishery developed further, it progressed, and this is kind of the classic progression of a fishery across the stock range. The fishermen started going farther to achieve the same catch rates or similar catch rates, and or the technology and those things improved. And you can see that sequential movement from region 2 into region 3 in the 20s, 30s, 40s. And by the 50s and 60s, there was roughly equal catch in regions 2 and 3. And then in the 60s, you start to see region 4 and 4B coming into the picture. And by the 2000s, we had catch distributed across essentially the entire geographic range of the stock. We had the proportions. I've got a similar figure somewhere that actually plots this proportions, and you can see that it goes from basically all region 2 to equal proportions And then we have this oscillation between fishery, a lot of fish in region 2, a lot of fish in region 3. And we've had 2 periods of much lower yield from this stock, one in the 1970s and one in the current period. Had about the same amount of yield in both of those periods, with about the same spatial distribution, perhaps a little bit more yield in the last couple of decades in region 4 and 4B than we had in that period in the 1970s. But we've had similar levels of productivity in both of those. I'll talk more about the PDO and the potential for that to be linked to environment in a few minutes.
To the intent.
Sure.
This looks a lot like Atlantic cod in the northeast US, sort of special depletion stock components. And there's pretty good evidence that it's historical contingents, subpopulations. Is there anything like that people talk about here? Places that used to have a spawning aggregation that got wiped out.
Possibly. There is some evidence, for example, of some spawning off the west coast of the U.S., the lower 48. We've not, and actually still some anecdotal reports of continued observations of fish spawning off the U.S. west coast. But we don't, I would say we don't have any direct evidence other than that, perhaps, of areas that had historical spawning that no longer do. It doesn't mean that there isn't, there couldn't have been a finer scale population structure at some point that we didn't detect. But I don't think we have any components that are clearly missing from the stock, the way I think they showed, at least in the Gulf of Maine, there were entire areas that just disappeared. That this area off the U.S. West Coast is something I know Joseph's done at least 1 or 2 proposals to try and look more fully at that. The challenge on the West Coast is that there's no juvenile halibut within 700 or 800 miles of the Washington coast, even though the habitat looks great. And so we've never seen a halibut, say 3 years old or less in that area. We see a few off the southern end of Haida Gwaii, and there's a little bit of spawning there. And then we have some spawning aggregations farther north, but it'd be an interesting question to know if there were spawning off, say, the Washington coast. Is that a population sink? Where do those fish end up? Because the oceanographic conditions, there's a separation off Vancouver Island that generally goes south. So where would those fish end up?
I have no anecdotal reports. People historically catching time halibut.
Not that we're aware of, no. We occasionally get reports from the west coast of hey, I caught a whole bunch of small halibut, and we say, send us some pictures. At least in a couple of cases, they've turned out to be petrale sole or something that looks a lot like a small halibut. If we look at then, so this is just commercial landings, and up until the last several decades, the directed commercial fishery was the primary source of mortality for Pacific halibut. You see this graphic just shows you across all geographic areas the distribution by sector, and the green being the commercial longline fishery, which was the primary source of mortality until the foreign fleets showed up in North America in the 1960s. That's the purple. That's discard mortality in other fisheries not targeting halibut. Those fisheries had a pretty important effect on Pacific halibut through the 60s and 70s. And then in the 70s, we started to see the increase in recreational interest in Pacific halibut, as well as discard mortality after the mid-1970s when current minimum size limit went into place. And now we have a much broader spread with the reduced overall mortality and distribution across different components. The sources of mortality are more varied in the last few decades than they have been over most of the historical record. So if we look into the commercial fishery CPUE, this is the one series that we have that goes all the way back into the early 1900s. I think it's 1907 or 1908 is the first year of the time series, obviously highly uncertain. This is the first piece of data that the commission collected throughout its record. We took CPUE from the commercial fishery, how many skates, how many hooks, how much did you catch? So this goes all the way back. We had, this would be an incredible data set that gave us consistency across the whole thing, albeit through the commercial fishery, except that between 1983 and 1984, we had a revolutionary change in the fishery where they changed from J hooks to circle hooks and effectively doubled the catch rate overnight. We have stories of vessels exchanging boxes of hooks and $100 bills on the water. It was such a big development. It made a huge difference to people's catch rates. And so much so that it took a year for everybody to go from J hooks to circle hooks. And that's that big gap. So that wasn't a jump in CPUE. That was actually a change in gear. It corresponded, however, to a time period when we were seeing in the years leading up to that, we had been seeing the CPUE increasing rapidly. And then it continued to increase after that. But that does create a fairly large disconnect in the commercial fisheries CPUE time series. The survey time series is much shorter. So we have a little bit of spotty survey information from the 1980s, which generally reflects sampling only in the core of the stock, region 3, region 2, and 3. And then it wasn't until the 1990s that we have a geostatistical approach and we can actually capture the full range of the stock. So we have two things going on here. We have the change from J hooks to circle hooks in the mid-1980s, so that's flagged there, and that's the first big jump in CPUE. And then we have the spatial change from just catch rates in the core of the stock, reflected in those values in the late 1980s. I think it's 86, 7, and 8, or 7, 8, 9, 5, 6, and 7. And then these estimates at the end of the time series, starting in 1993, those are all from our current uses. So those integrate, those are estimates of numbers per unit effort over the whole stock and associated uncertainty. And you see how much broader the confidence intervals are in the beginning of that time period because we didn't have survey coverage over much of the coverage during that time.
Can you remind me how you deal with the gear change and the assessment? Is that just the offset? That's
Yeah, so we estimate an unconstrained deviation in catchability for this series in 1983 and 1984. So effectively, it's two different cues. We also have age information from the commercial fishery that goes all the way back to 1935. So this is also a pretty impressive record compared to what's available for stock assessments. You can clearly see some strong cohorts coming through. There's a cohort somewhere there in the late 1950s. There's another one in the mid 1960s. And the biggest one in the time series is that strong 1987 cohort, which you can see going through up through the age composition during that time period in the 90s and early 2000s. And that cohort was apparently the largest cohort in 100 years. I back calculated, just ignoring aging error, I back calculated that we extracted about 220 million pounds of yield from that single year over its lifespan. So that was unprecedented in the age composition going back for nearly 100 years. A couple of highlights here. You see, starting in 2017, we have sex-specific information, so males and females are separated in the information. Prior to that, we didn't have that. Of course, they're addressed at C, and they're virtually impossible to tell males and females. You can also see the effect of the minimum size limit that was put in place in 1973. Prior to 1973, there was a minimum size limit in the mid-1940s of 26 inches. It actually started out in terms of pounds, and they realized it was unenforceable. It was then switched to 26 inches, but it really didn't have much of an effect on the fishery because most of the fish less than 26 inches were not selected by commercial gear. Some of the literature suggests it was an effort to, it was sort of a backdoor allocation issue at the time to make sure that those small fish couldn't be retained by anybody else because it really didn't have a big effect on the directed commercial fishery. But the 32-inch minimum size limit did. Some of those fish less than 32 inches are selected by commercial gear. And so you see that the 3s, 4s, and 5s virtually disappeared when in the commercial catch when the size limit was imposed in 1973. And with the exception of a few 5 and 6 year olds, we generally don't see any fish less than about age 7.
Why is it that there is no fish older than 20?
Yeah, thanks for the question. So that there are fish older than 20. I've used 20 as a plus group up until 2002, and that's because those are the years when we were using surface aging. So fish older than 20 we know are fairly biased. There's a slight bias in fish older than age 15, but it becomes much more pronounced the older they get. So in the model, we both account for that bias. So we've got an estimated aging imprecision and bias vector that we use to adjust these ages when we fit them in the model. But we also aggregated age 20 over that time period to avoid trying to draw those older fish. But you can see that even in the plus group, there's very few fish during that time period.
Is that something you developed or is there a paper?
There are several papers on that. The imprecision, yeah, there's Bill Clark did some of the initial work on that, looking at bias and imprecision, and then we've refreshed some of that information. One of the things that we did about 13 or 14 years ago now was to look at whether or not some of the changes in size at age could be related to drift in the aging method. It's kind of Occam's razor suggested that maybe we haven't seen such big changes in size at age. Maybe we've actually just had a drift in our aging method over time. So we went back to the archives and we re-aged 500 otoliths from every decade all the way back to the 1920s. And then we compared them to the ages that were read at the time, and we re-estimated the aging factors, and we found that the aging is actually remarkably consistent. Yeah.
No, I meant the fitting of on burlap. Sorry.
We actually don't use a growth curve. Yeah, we just use empirical weighted age. So we take we have the estimates of weighted age, and we use the models all run in numbers at age, so we don't even have to go through. Which is good, because when I show you the growth curve, you see why we don't fit. So to summarize the data that's available, we have, it says catches, but it actually should say, well, I guess catches, right? It's actually mortality for non-directed discards. It's not the total catch, it's the part of the catch that we assume to have died from the capture. For those fish that were discarded and survived, they're not included here. As I've shown you, we have abundance indices that go all the way back in time, noting that we have primarily region 2 and region 3 prior to the 1970s and 80s. We have a little bit of age composition going all the way back, but it gets more and more informative as time goes on. And likewise, we have that information for both the commercial fishery and for the survey. There are some tables and figures both in the assessment document, and we also produce a separate overview of data sources because there's so much information to provide. Rather than pack it all, many assessments will put all the data figures, all the modeling into one document. We actually break it into 2 documents so it's easier for you. So the synopsis of this is that we have a pretty good look at sources of mortality all the way back through this whole time series. The soft spots are that there's uncertainty in the discard mortality rates. So for discards, for non-directed discards, for some part of the recreational fishery, there is certainly uncertainty in what fraction of the fish that are discarded subsequently die. And that's not currently captured in any quantitative way in the stock assessment. We also increasingly recognize the importance of whale depredation, and that's a source of uncertainty that's not included quantitatively in the current stock assessment. I've done some sensitivities, many sensitivities to that over the years. You'll remember from a couple of years ago, I did a lot of work on whale depredation from logbooks and observer data. We can speak more to that later if you want. Primarily have just fishery data before 1992. So the long time series is primarily being informed by fishery dependent information for all the challenges that poses. We model, as you'll see later in this presentation, we model time varying catchability in the stock assessment. It's a random walk in catchability. So things are allowed to drift, technology is allowed to drift over time. But of course, when you go way back in the time series, if there's no other source of information, then you're still going to get the trend that you're getting out of the fishery, even if you look for some random walk. We have really high quality age data after about 1992, when we have all the areas and fleets and everything represented. But we have this change in methods from surface to break and bake ages in 2002. And so it takes a little extra modeling to deal with the ages for the first 10 years of that time period. And we have very limited biological information on the discards from the directed commercial fishery, from the recreational sector. We don't have anything on the biology from subsistence. And the non-directed discards, while we have length frequency sampling in many of these fleets that encounter halibut, that's it. So we have to rely on trawl survey data to create an age length key, to turn those lengths into ages, deal with that mortality and stock assessment. We have a number of other inputs beyond the raw data that go into the model. And they're not treated as data, but they're generally treated as if we know what they are. And so this is what they are. There's a table 8 document that outlines its various sources. The first is the Pacific Decadal Oscillation Index, and that was updated this year. I'll talk quite a lot more about that. We treat that as a covariate to the stock recruit function. The next is the maturity ogive. You just heard the presentation about the background on that. Again, we've updated that this year, and I'll show you not only the results of updating it, but also some sensitivity to updating it and dealing with truncate or don't truncate the ages. We have, as were developed in the last, in the 2022 stock assessment, we have priors on natural mortality. For adult fish, we also have a decreasing function where natural mortality is elevated for ages 0, 1, and 2, and then assumes a constant lower level for ages 3+, consistent with kind of the Lorenzen concept of increased mortality at small sizes. Interestingly, for halibut, by the time they're 3 years old, they're as big as basically all the other flatfish in the North Pacific. So that there's extensive amount of background on that in the last full assessment in 2022, where we did a meta-analysis across all the flatfish in the North Pacific.
Yeah, I can't remember seeing a picture of the priors and the posteriors. How well estimated is that on
Yeah, I didn't re-include that figure. I don't think in this assessment. The priors and the posteriors were available in the 2022 assessment. For the models we estimated, it's reasonably well estimated. We don't estimate it in the coastwide shore. We don't estimate natural mortality for females. We do estimate natural mortality for males. I have a table coming up that has the confidence interval for the estimates, so we can look at the range that's suggested from the estimates as well. As Colin noted, one of the important assumptions we make is that fecundity is assumed to be proportional to body weight. We've done some back of the envelope looks at some of the data sets we've got and haven't seen anything to suggest otherwise, but until we get actual fecundity estimates, this is an important assumption. And certainly we know for species like cod and rockfish, there's a lot of evidence of non-constant reproductive output per body weight. So it's definitely something that's a possibility for halibut, but that's our top research priority after as we're completing maturity right now. And we actually, I'm sort of to preface some of the discussions we've been having, we even had some discussion about whether we should update the maturity until we have fecundity. Should we wait and do both of them at the same time? Potentially, we've got maturity shifting to the left. If we have a fecundity relationship, it's going to shift that back to the right. Should we be doing both at the same time? It's certainly an open-ended question, but we don't know what the result of that analysis is going to be. We calculate weighted age external to the stock assessment model. That's something we've been doing for many stock assessments. And the quality of those estimates varies with the portion of the time series. Some of it is reconstructed from previous reports early in the time series. And then I'd say over the last 30 to 35 years, We have some pretty incredible weighted age estimates. We sample on the order of 10 to 20,000 fish a year for age and weight, age, length, and weight. So we're very well informed when it comes to what's actually happening. The challenge, of course, is what's coming, what was driving that, and we have really no mechanistic understanding right now. We use a length-weight relationship, and that's primarily to convert weights and lengths for fisheries where the only lengths are being collected. I've talked about the aging error, bias, and precision that's used in the model. We have two matrices, surface aging and break and burn, and those are treated separately for the data sets that use one or the other. We also have what I'll call priors on bycatch, discard, and recreational selectivity. Historically, we would just assume a selectivity curve for these sources. What we do now is we use, for example, for discards in the directed fishery, we use the biological information for sublegal fish that we capture on the survey, and we fit that in the model, but we downweight that. So essentially we're able to estimate selectivity, but we're not giving the model information on recruitment. It allows us to propagate some uncertainty in the estimates of selectivity associated with these fleets. So I'll talk more about those approaches.
Is the tagging data used to estimate the recreations?
No, we don't have extensive enough tagging information to do that. So we have spot tagging projects in a few areas, but not the recreational fishery occurs from California all the way to the Bering Sea. We just have a few little spot projects. What we have for the recreational selectivity, we have a pretty dedicated sampling program that goes on to the Gulf of Mexico, and they sample quite a few fish from the charter fishery. And so those age compositions are used to recreational selectivity. So similarly, we fit them in the model, but we downweight them substantially. So we're not getting strong recruitment, just getting and those age comps, actually, they catch fish in the 2, 3, and 4-year-old range. So we see they're generally, I don't think I have a figure here, but in the data overview, I have a figure, we can call it up later. We generally see the same year classes that we see in the other fisheries. We just see them a few years earlier in that sector. If we had comprehensive sampling across all the recreational fisheries, it would be potentially a pretty informative data set. But we don't, there's no mechanism in place to collect age data from the recreational fisheries currently, except in the charter fishery in 3A and to a small degree in the and only recently in the charter fishery in 2C. Everywhere else just collects lengths from their recreations.
So these 60,000 tag returns that you referenced.
It's spread across research projects, but primarily commercial fishery. Many of those tags were put out using other methods, but the primary source of recovery has always been in commercial fishery. We get a few back in the survey. We get the fish from other fisheries or from the recreation sector. Historically, the vast majority of our tags have come from have been recovered. So just to orient you generally to the management of Pacific halibut, as I mentioned in the composition slide, we do have a 32-inch commercial minimum size limit that's been in place since 1973. We have commercial fishing seasons. Generally, these open in March and close in December. That's a slightly longer period than we had, say, before COVID. Things were extended a bit to provide people more flexibility. Historically, we've had fisheries that often opened in April and closed in November. So those have been extended a little bit. The challenge in Alaska is that the halibut and the sablefish fisheries are linked in terms of their starting and opening dates. And with very large quotas for sablefish in recent years, there's been a lot of pressure to leave the fishery open longer to give people time to catch all their fish. So the concern would be if we went to a shorter season for halibut, it would limit the sablefish fishery, and they're linked so that harvesters that are catching both species aren't discarding one so that they can then catch it later in the season, vice versa. Importantly, our survey occurs primarily between June and August, sometimes a little bit in late May and sometimes a little bit into early September, but primarily just during the summer months. So when we look at many of these data sets, we do need to remember that the commercial fishery is representing most of the calendar year, and the survey, like many do, is representing a snapshot that occurs in the summer. In Alaska and in British Columbia, it's an IFQ or ITQ fishery, so quota fishery where the harvesters can choose when and where to harvest their fish within where they own quota. It is still a derby fishery. The commercial fishery in Washington, Oregon, and California generally occurs in a couple of openers, sometimes 2 to 3 days long, usually right around late June or early July in recent years. And that's, while it is not open access, you do need a permit. Basically anybody that wants a permit can get one and participate in that fishery. So it's effectively open access, although it is a permanent fishery. They know how many vessels are going to go out and fish at the time that they open the fisheries.
Is there any like frequency change in the commercial fishery over that seasonal period? Is this period representative of the comps?
There are a number of really interesting patterns in a commercial fishery. Seasonally, it really depends which parts of the commercial fishery you look at. It varies geographically. Some fisheries, like in the Bering Sea, there's not a lot of fishing that occurs until late May or early June, where in the Gulf of Alaska and down through British Columbia, lots of harvesters begin as right on the opening date. Within certain geographic areas, we see some very distinct strategies. Sometimes early in the year, there are trips that target deeper areas that apparently look like migration corridors. They catch larger deliveries of generally smaller average size fish. And then in summer, there's a distinct harvesting strategy, which is inshore fishing targeting larger fish, generally smaller deliveries. These are people often who own smaller amounts of quota and have the luxury and or ability to maximize the value of that quota by waiting and going to areas where they're likely to encounter larger fish, which on average will give them a higher price, although that's less than recent years. So there's definitely some very distinct harvesting strategies across the fleet season. There's nothing that I would say is consistent across everywhere. It's not as simple to say they catch small fish in the spring and big fish in the summer, but there's definitely some. And year to year, I think that can an effect on our composition data, particularly if you look at the level of individual regulatory area. But it tends to wash out at the longline and pot gear is legal for Pacific halibut. So harvesters that catch halibut with those gears may retain halibut if they have quota to do so in those areas that are quota fisheries and or it's the fisheries open. Trawl gear has and always have to discard all Pacific halibut they encounter. So all of the trawl fisheries that occur in the North Pacific, US and Canada, when they catch Pacific halibut, they have to discard that. Those discard mortality rates range from somewhere in the 50% range up to 100%, depending on the conditions of the fishery, where and when they were caught, how they're handled. There's a pretty extensive program in Alaska now to allow for deck sorting, which is also the same approach that's used in British Columbia and Washington, which is that they tend to sort the catch immediately on deck, the halibut go back over, and those fish tend to survive at a much higher rate. If discarded within the first 20 or 30 minutes, the discard rate can be, like I said, down in the 50% range as opposed to up in 80 or 90% range. Even after 30 minutes, it's pretty remarkable that this fish can last. The recreational personal use and subsistence is managed by the domestic agencies in both the two countries, and it differs by regulatory area and by year, and it would take all day to explain, to give you the history of all of those changes. There are size limits, bag limits, annual limits, possession limits. The regs are different in BC than they are in Southeast Alaska. It's incredibly complicated. And as Claude mentioned, it's about 15% of the overall mortality comes from recreational fishery. So there's certainly a lot of change over time and change across areas going on in that fishery that we don't try to capture at all. We're not trying to have time varying selectivity to capture any of that. We're just trying to get an aggregate selectivity for that component. That's it. And as I mentioned, aggregate discard mortality rates range from 4% to 100%, depending on the fishery components. As Claude mentioned, in the tiny niche of the excellent condition recreational fish, it might be as low as 1.3%. And in the trawl fishery, where the fish are caught, put in a holding tank with tens of thousands of pounds of other fish, and then run down a conveyor belt before they're sorted and put out a discard chute. Okay, it's going to be 20 minutes before lunch, so why don't I tackle a couple of these topics, and then we'll be well situated to get through the remainder in the afternoon. So I'm going to focus now on some of the changes for this year's stock assessment. The first is a bootstrapping approach to age composition data. So in the 2022 assessment, we introduced a bootstrapping approach to estimate the effective sample size of the input age composition data to the stock assessment. This was based on some work that I did with Owen Hamel back in the early 2010s, published in 2014, and was recently extended by Pete, who's online, and Ben Williams at the Alaska Fisheries Science Center of NOAA Fisheries, to include the concept of including aging imprecision in that bootstrapping approach. So what this basically does is it says, based on your sampling design, how good could the information in your age composition be? And so it's a resampling with replacement of your sampling design. And you compare the age composition you got to the distribution of age compositions you could have gotten through that resampling. And what you find out pretty quickly is that your nominal sample size is vastly larger than your effective sample size because of your sampling design and all those other things. And the extension that was added by Hulson and Williams was to say, well, don't forget about the aging imprecision. So when you create your age comp, it's only one read of each otolith, but you could have, hey, it comes from a distribution of possible reads. And so it adds a resampling of the aging imprecision matrix as well. So, and I think this is a nice extension of that earlier work. So this is actually the aging imprecision matrix from our break and bake ages. And you can see that and if your eyes are really good, you can actually read the numbers. If they're not, you can just see that things not surprisingly line up pretty strongly on the diagonal. But there's quite a few ages that are off by a year or two or three or four or 5, right? Yeah, Sean?
So does that mean you don't use this in the likelihood anymore?
No, we do use it in the likelihood. This is only to determine the input sample size in the model. We still use this to, we still have an aging imprecision matrix when we fit the age data in the model. Because when we fit the age data in the model, we have the true population. And then to create an expectation from that, we use this to generate an expectation, which is compared to our observation.
Which accounts for the aging.
Yes.
So why, why it's being used here then?
Because this is just to determine the sample size. It doesn't affect the age composition itself. It only determines the effective sample size that we're starting. So this is basically saying, what's the best precision we could have gotten from the input sample size or from the sampling design, conditioned on how many fish we took from how many trips, from how many strata, and the potential for individual reads.
Let's see why the aging error has to be included. It's not part of the, you're not sampling conditional on age. Age is a part of the sample.
But each age is only a single realization from
Right, but you're accounting for that in your likelihood.
This isn't affecting the age composition you put in the model. It's only affecting your maximally possible sample size.
Which is a measure of the variance of the.
So it's not affecting the expectation, only the variance. And that's a component that you don't get in the stock assessment. That's only affecting the expectation. So one method is dealing with the different expectations, and this is dealing with the difference in variance associated with aging and precision. So Pete's papers, it's a good read. I would encourage taking a look at Pete's paper. He provides some more background, and they apply this method broadly across a wide range of species in Alaska.
Variance is a fixed input to the stuff, so into that likelihood. That's why it's useful.
So we have two of these matrices, one for the break and bake method and one for the surface aging method. They're both in the document. I think I only have one here in this presentation. The result of this is, not surprisingly, a smaller effective sample size than you get without it. So in the 2022 analysis, when I bootstrapped only the sampling design, I was getting somewhere on the order of an effective sample size of 4 per sample. And here a sample would be a trip or a set for the survey. In this case, it's a somewhat more variable relationship, and it's more like 1.5. So it definitely suggests a lower effective sample size with more variability. And a lot of that variability comes from the historical samples, which are the surface aging method, because that's much less precise than the break and bake method. So again, this doesn't affect the age compositions that go into the stock assessment, only the sample size associated. That sample size only serves to create a maximum. So this is the best you could do under the sampling design you had. The true sample size is going to be something less than that. It's going to account for the lack of fit in the model, as well as the observation error. So it's really just the observation component that you can quantify through this external booster. So it's a maximum input that you can then work backwards from samples. So we've added that here, and I'll show you the effect of that down the road on the stock assessment model. But I think that was a nice extension to the earlier work, and it makes sense. The work that I originally did with Owen Hamel, we were actually doing this for length composition data, so we didn't have this to worry about. I suppose you could do it for whatever imprecision you have in length measurements, but for most species, that's you're binning and taking care of that. So this really is specific to age data. So nice extension there. You're able to apply it in the assessment. I think maybe I'll do one more section here, maybe 2 of them before lunch. I don't know. I think our lunch is here, so might as well keep going. Okay, the next piece would be the Pacific Decadal Oscillation. So this is an index. This has been found historically. We've known for approximately 25 years that the PDO is strongly correlated with average recruitment time period. So this is the PDO anomaly that this is the index that we've been using in the stock assessment. And here I've highlighted in green and red the stanzas or regimes in the PDO. So we had historically estimated a high PDO regime up until the late 1940s. Lost my, hi there. Oh, there we go. So we had a high regime to the 40s. Then we had a low PDO regime through the late 1970s. And as you remember from the yield graphic I showed you, that corresponded to that time period. By the end of that, we were at the lowest yield we'd ever seen. Then we had a high period from the late 1970s through somewhere in the early 2000s. Then we've had kind of a mixed bag since then, a little bit up and down. Again, this was, I didn't invent this. This was determined in the early 2000s by Bill Clark and others. That the PDO itself dates back to 1997. Nate Mantua first identified this broad pattern. It's not a single index. It is a proxy for productivity conditions in the Gulf of Alaska. It actually represents a sea surface height anomaly, but it is generally regarded as a proxy for lots of things. The circulation pattern in the Gulf of Alaska, the temperature, the sea surface height. Positive PDO generally corresponds to a counterclockwise gyre in the Gulf of Alaska at warmer temperatures, which is good if you're a groundfish species that spawns on the slope and needs to get inshore and to the west. And it's good if you're a species that benefits from slightly warmer water in the Gulf of Alaska. it's got a reverse relationship with the West Coast. So when it's warm, it's, for example, some of the early relationship was determined for salmon, and when it's warm on the West Coast, it's bad for salmon at the southern end of their range. And so it's an inverse productivity regime where bad for salmon on the West Coast, good for halibut and salmon in the Gulf of Alaska when it's positive, and the opposite when it's negative. So this relationship's been around for a while. It's been recreated or refreshed and added to several times. But there's a new version of it out there. So a few years ago, the analysts that create this time series went back and recreated it all the way back into the mid 1800s and standardized the methods across the whole thing. So the previous series that we've been using actually had a couple different methods spliced together. They were using a slightly different approach to create the anomaly in the most recent period than they were for the historical period. And so they've proposed this new method, and there are links. You can go in the document. There are links to the data repositories that provide this information. So the longer time period, though, is also standardized to a different period of average years. So it's an anomaly, but it's an anomaly to a slightly different average. And so you can see, if you look back and forth between these things, it changes the junction point between some of these regimes. It's also, of course, the x-axis is different, so it looks a little different. You still have that period of low productivity from the 40s to the 70s. You have a high productivity period from the 70s to somewhere around 2000. It ends a little bit earlier, and then it seems to be much more strongly negative in the late time period, or at least relative to the average than we had in the other time period. The definition of a regime that we're using here is at least for 4 sequential years of one anomaly or the other, either positive or negative. These are annual values. There's actually, they calculate this by month. So this is
And have you played around using the actual anomaly?
Yeah, I have. So in the 2022 assessment, I looked at 5 different hypotheses. One was using the annual anomalies. One was using just the biggest anomalies to see if this was really more of an index of windfalls rather than everything else. And the short answer is I couldn't find anything that worked better. And I didn't want to look at so many hypotheses that I was guaranteed to find one mentioned. So I kept it to a fairly limited number of hypotheses, and didn't do any better than the original relationship. And I would say, to their credit, I believe that Bill Clark and Stephen Hare did something similar in the early 2000s, which is that they tried to come up with a few hypotheses. And the leading hypothesis is that this creates favorable conditions and transport for eggs and larvae. That when the gyre is going counterclockwise, if halibut are spawning on the shelf slope break, they need to get inshore and to the west. That's where most of the juvenile habitat is. And so survival and transport seem to be good when the PDO is going in that direction and the water's warm. And when it's going the other way, one would assume most of the advection is south and offshore, which can't be good if you're spawning on the shelf slope break and you're needing to get to the Western Gulf. Most of the juvenile rearing habitat for halibut is west of Kodiak and into the Bering Sea. And the work that Lori Sidoris and Ray Webster have done have shown that the oceanography will take fish from even as far away as the Central Gulf to the west through the Aleutians and into those areas under the right transport conditions. Actually, it'll take them as far across as Russia, which is why I think there probably is some demographic links to Russia.
In that case, what about using current vectors from the ocean?
It would certainly be a possibility. If we had an oceanographic modeler here who wanted to jump on that, I'd love to try some of that out. There are some series available, and it's something we could investigate. I know there are a couple of there's a PAPA trajectory, and there's a couple of different drift models for the North Pacific that could be evaluated. It's not something beyond the PDO that we've spent a lot of time doing here, other than the work that Lori and Ray published a couple of years ago. We can provide that reference if needed. So the decision point here was, okay, we've got this old series that's not strictly comparable across the whole time series.
But it is the series that the relationship was developed on. And now we have this new series, which changes the definition of the regime slightly, but does give us a longer perspective. You see, it has actually somewhat different perspective prior to the 1920s. Instead of just having a long period of positive residuals, it's actually got a negative period and then another positive period and a negative period. So it's a little bit different perspective on, especially the historical component. And it's a little bit different in the more recent periods, suggesting a fairly long negative period here at the end of the time series with this thing. So the decision point here was scrap this whole thing, because do these things even work in the long run anyway? I mean, the fisheries is littered with examples of relationships like this that work for a while and then fall apart. Right? And there's just the sardine example. I mean, there's just all these, but nonetheless, every year that I've been here, we've re-estimated this relationship, and the coefficients have remained pretty similar the whole time. Now, to be fair, we have age data that starts in 1935, so the vast majority of the data haven't changed even since Bill and Stephen did this work in the early 2000s. They already had 70 years of data, and since then we've added another 25. But the relationship, the coefficients, if I were to plot the coefficients estimated for this relationship over the last 15 assessments, it's not like they're trending slowly down. They bounce around a little year to year, but they've stayed pretty consistent. So there seems to be something useful here, is my conclusion. The decision point for this year was how to make the transition to this new time series. And I think I left you with a cliffhanger here. Good job. So I'm going to give you all this background, and then later on today, I'm going to show you the results of this. Sorry about the cliffhanger. Maybe I did that on purpose. I was trying to stick with the data before we dove solidly into the modeling. So I am later on going to show you the transition from the previous series to the current series, and an evaluation of the coefficients, the amount of variation explained, and the effect on the stock assessment. Okay, although lunches aren't here, right? Might as well keep going. Okay, you already heard, this is a quick section, you already heard from Colin this morning about all the background of our data collection and analysis of maturity. So for the stock assessment, I boiled this down into three different potential avenues here. One is we could stick with the current curve, which is the curve that's here in black. And I'm sorry, I used different colors than Colin did, so don't try to line up his colors with my colors. The black curve is what we've been using historically in the stock assessment. And as Colin noted, that's based on data from 2002 to 2004. 2002 to 2003 from 2B, 2C and 3A.
It's 2B and 3A.
2B
We thought
and
it was
3A.
2C, but then we corrected it.
Thanks. 2B and 3A. So a small amount of the stock on a fairly limited time period. The reason they didn't try to go back farther is because they were worried about the bias in ages from surface aging. So they stuck to only the couple of years where they had break and bake unbiased ages. And that was generated for the 2006 stock assessment. We've not updated that, and that's a single curve that's used for the time series. So that's our best estimate of the maturity schedule for Pacific halibut. We now, with several years of histological data, we have the curve that's here in red, which is what Colin just described for you. And that's the original curve was truncated at age 8 because they had never observed a fish younger than age 8 that was mature. Now we've truncated at age 7 because we haven't observed a fish younger than age 7 that's mature. The little green tail there is the extrapolation from the GAM model of what could be the case if you believe that fit. And I included that here. We were uncomfortable just putting that in because we don't want to be creating mature fish where there's no biological evidence for it. And similarly, there's potentially a lot of fish in those age categories. So a small percent mature could add up to a fair amount of spawning biomass. As you'll see when I get to the sensitivities, it's kind of a non-issue, but we want it to be explicit and transparent. And so all three of these are fit in the stock assessment. compare the results. The key takeaways here, before I even show you those results, are if you just look at the red and the black, you'll see two things. One is that the red is shifted to the left. So as Colin showed you, the L50 is 11 years instead of 11.6 in the black. But at the high side, the red curve is actually a little bit lower. So what this is saying is that we're getting more spawning biomass from slightly younger fish and less spawning biomass from slightly older fish. So if you think about how that's going to affect the population dynamics, what it's going to mean is that we're going to have less biomass when the stock is large and more biomass when the stock is either small and dominated by young fish, or when we have recruitments coming in that create a lot of young fish. So it's going to change. It's going to flatten out some of the dips a little bit. We would expect it to flatten out some of the low spots in the stock a little bit if we apply this for all other things being equal.
So Colin's results suggested that during Jordan.
Yeah.
A future goal.
It is. So I think our takeaway was we certainly see evidence that this stuff could be changing over time, but with only 3 years of histology, it's difficult to know the difference between process variation in maturity and just observation error with where we had samples in those 3 years. We had different timing, different geographic layout, and it's only 3 years. So we don't know if it's just interannual variability or it's actually some coherent trend.
Sure, if you restrict it to the histology,
Yeah.
individual stuff, that Colin should pretty.
It seems to show a trend of increasing maturity and then decreasing maturity again. Yeah. And so functionally, it would be very straightforward to add that, to add time varying maturity to the stock assessment. The way maturity affects technically the stock assessment, we have a matrix of weighted age, and it's a multiplier on that matrix to give you spawning biomass at age. And it's a vector multiplier. It could be a matrix. It'd be easy to have it be a year specific. We don't have, of course, information prior to the 2000s, so we would still have some constant other average. So our conclusion this year was that we should introduce the new maturity information, and then perhaps when we have a geostatistical model and a little more analysis of the time aspect of things, think about moving to a time varying maturity approach as well. And or when we have fecundity, that's then a second multiplier on that same matrix of fecundity at age. Again, that could be either a single vector or it could be a full matrix of fecundity at age of that term, but that also varies over time.
That's not something that.
But we won't be able to back calculate that either. And we don't have anything like a visual estimate to calibrate a historical term.
Yeah, we wouldn't be able to recreate a time series like we have for the visual. Yeah, on fecundity.
But at least you'll know.
Yeah.
And algebraically, as Colin was saying, there's some conceptual thinking about whether you include, say, skip spawning or, skip spawning is kind of a binary thing. As Colin noted, it's really more of a scale. There's atresia, there's 10% atresia and there's 90% atresia. And you can algebraically, for me in the assessment, it makes no difference whether we include it in maturity or fecundity. They're both multipliers of the same matrix. So it's more, how does it make more sense to model it biologically, and how are our data collected in such a way that we can most reliably estimate it? And then however that is, put it in the assessment, whether it's a change to maturity or fecundity is not. That's a natural break point, and it's 12:30. So I'm thinking maybe we take a break, and then it's a pretty good point to move into the evening afterwards.
Good. Thank you. We're going to take a break until lunch break. Till 1:30.
Oh, watch this.
Yeah, maybe longer.